Dr. Fatma, please go ahead. You are now live.
Okay, Bismillah ar-Rahman ar-Rahim. Dear guests and colleagues, Assalamu alaikum, and a very good day to you all, wherever you're joining us from, whether it be inside or outside Qatar. My name is Fatma Darak Shan Rassam, and I'm the Biomedical and Health Manager at the Qatar National Research Fund, commonly known as QNRF, the primary funding agency in Qatar. So before I begin, please note, since this is a virtual seminar, we are naturally prone to technical challenges, as we have had early this morning, and possibly unexpected fire alarms, emergencies, etc. So please, apologies to all beforehand if you face any difficulties, and we'll try to resolve them as soon as possible. So on behalf of QNRF High Management, the Biomedical and Health team and myself, welcome to QNRF's Biomedical and Health Research Outcomes Seminar with a focus on the very important topic of research in pediatric diseases and conditions. Now, the aim of this seminar is to highlight the achievements of the talented scientists in Qatar and showcase their great work and the advances they have made in pediatric research, which has been funded and supported by QNRF over the years. The seminar will showcase not only the expertise, but also the resources readily available in Qatar, which now attracts international collaborations, something which was not conceivable 15 years ago before the inception of QNRF. As you are all too aware, the COVID-19 pandemic in the preceding year challenged all aspects of healthcare in all countries, in every location. But thankfully, medicine in the 21st century is far different to that it was a century ago. And within weeks, the RNA sequence of the pathogen was fully decoded and published, its genetic evolution captured and published rapidly. In fact, within months of the outbreak, testing of the first in-human vaccines by pharmaceutical companies began. The number of studies related to COVID-19 has since increased daily. The scope and magnitude of engagement was and is striking, and the extent of global collaboration unprecedented. But the pandemic not only compounded all challenges, namely the strain on our healthcare system, but also created new challenges. The pandemic shortened most academic, industry and governmental basic science and the clinical research in children's diseases globally, all redirected research to COVID-19. Research which was fundamental to the progress of children's health prior to the pandemic. We at QNRF and the researchers we support, both inside and outside Qatar, stepped up to these research disruptions in an attempt towards restoring normalcy within pediatric science. Furthermore, as the following presentations will show, we at QNRF remain committed to strengthening our national research capabilities to respond to local health needs. An important goal for QNRF is not only to help develop more scientists, but also pediatrician scientists who are dedicated to advancing the application of knowledge gained from basic science to the health problems important to children in Qatar. It is now more important than ever that child health research remains a priority in Qatar's national agenda. By hosting this event, we hope to raise the profile of pediatric research in Qatar, also for the research community to engage in discussions to reach decisions regarding prioritization of long-term funding goals for dedicated pediatric research beyond this forum. And the lessons learned from the pediatric research during this pandemic. We are resolute in our commitment in supporting you to continue the progress of research to meet the needs of children, especially those affected by preventable diseases. So before we make a start with the speakers, please allow me a few minutes to provide some data and statistics of QNRF's funding support in this field. If nothing else, because we are dedicated to statistics. So I don't know if you can. Allow me a few minutes. Can you see my slides by any chance? What?
Yes, Dr. Fatma, go ahead.
The slides are available. Great.
Yes, go in a presenting mode and go ahead. Perfect.
Okay, so this is my, please reach out to me if you will. This is my email address, and you're more than welcome to log into QNRF's website for any further details. So let me start by the first slide. So as you can see on the left-hand side, I'll try to use my mouse. These are the awarded projects in pediatric research per research type. So as you can see, 44% of our research in pediatric fields has been in applied research, and around 29% has been in basic research, and around 27% in translational research. Now, of these, how many have been in which particular program? And as you can see on the right-hand side, the vast majority, that's 42% of the projects that we funded in pediatric research has been in the MPRP program, followed by 21% in the Europe, which is one of our capacity building programs. And there's also 11% in other capacity building programs. But you can see 12% of the projects we funded has been in our NPRP X or in NPRP exceptional. Here we have just broken down for you, again, breaking down the statistics. Where do these particular types, or as I said previously, these research types, where do they fall with regard to institutes? As you can see, 25% of the funds have gone to Hamad Medical Corporation, 22% to Sidra, followed by Qatar University. We've got Welland Cornell, Hamad bin Khalifa, and some of the other institutes. So these are the top 5 institutes have currently received the majority of the funds related to pediatric from QNRF. But as we know, now that the baton has moved from HMC to Sidra, if you like, and the vast majority of the pediatric units and divisions within HMC have now moved to Sidra, the expectancy would be that these number of funding in Sidra would increase. But we also recommend and encourage Hamad bin Khalifa and Qatar University and Welland Cornell to also reach out to Sidra in collaborations inside Qatar. And now the next slide will try to show you the type of research that each of these are the top 5 institutes that have been awarded funds have placed their efforts in. So I can see with Hamad bin Khalifa, 45% of their funds has been in applied research, 22% in basic, and 33% in translational. And then similarly as we go across the field. Now, with regard to Sidra, as you can see, the vast majority, 54% of the funds have been in translation research and only 10% in basic. Now, with Welland Cornell, 55% have been in applied, 36% in basics, and 9% in translational. So again, you are encouraged to collaborate, and if you feel that one particular institute has higher strength or bigger applications in translation, for example, you may want to reach out to them. And similarly, Sidra, if you feel that, for example, Hamad Medical HBKU or Welland Cornell have been particularly funded in basic science, you may want to reach out to them in that field also. For my very last slide, just to break down even further, which particular topic and field have the research areas with the funding been placed in? You can see that 16% of the funds have been placed in human genetics. You may just want to ignore the pediatric. This is just one particular area, but this incorporates all other research fields. And you can see this is just to highlight that we in QNRF are dedicated to funding all across the various research fields. Now we have, for example, microbiology, whether we have virology or allergy example. For example, these are the fields that you have shown interest in, and we likewise have funded. We also have, for example, psychiatry and psychology, which have also been funded, although in low numbers. But you can see from there which, if you like, are the undertapped areas of research. I won't give you any more statistic. I don't want to overload you, but if any of the institutes would like to have a breakdown of where exactly the funds have gone and further details with regard to your institute, you're more than welcome to reach out to me, and I will try to find out to give you further information. With that, I'd like to thank our Executive Director, our Deputy Executive Director, and our Senior Manager at Biomedical Health, and also the communications team at QNRF for all their help and assistance. Okay. We'll move on to our first speaker, and if you're sitting comfortably, we'll move on. For our keynote speaker, we have a pleasure to have Dr. Barbara Frieden, who is the Chief Executive Officer at Sidra Medicine, a women and children's specialist hospital in Qatar. As the CEO of Sidra Medicine, Dr. Frieden is responsible for overseeing the strategy and growth of Sidra Medicine healthcare, and most important to us all, the medical education and the research services. Dr. Frieden has extensive research in both the public and the private healthcare sector in Sweden and the Middle East. And prior to joining Sidra Medicine, Dr. Frieden was the CEO of Sheikh Khalifa Medical City in Ajman, and before that, the CEO of Schweiglenska University Hospital, Scandinavia's largest teaching hospital. Dr. Frieden sits on the board of a variety of governmental and commercial boards related to healthcare, including the leading medical medtech company and the Spine and Sophiskynet, a non-profit royal healthcare provider that conducts education, healthcare and research. Dr. Frieden received her medical degree from Umeå University in Sweden, specializing in obstetrics and gynecology, subspecialized in reproductive medicine and IVF. She was also a visiting research fellow in reproductive medicine at the University of California. She holds a PhD in medicine from Gothenburg University. A very warm welcome to you, Dr. Frieden. Pleasure to have you here, and thank you for your time. The floor is yours.
Thank you, and I will start by thanking the organizers at QNRF for inviting me to this beautiful seminar on outcomes, which in itself testaments to the good collaboration that is between education, research and medicine here in Qatar, and where QNRF plays a very vital role. So thank you for that. I will start by saying that every kid counts. And every kid in Qatar will, at one time or another, come through the doors of Sidra. These two sentences describe both the philosophy at Sidra and a huge opportunity that we have as sole providers of tertiary pediatric care in Qatar. Our philosophy is simple. It's to provide the best possible care with the best medical outcomes to each one of our young patients at all times. If we do our job properly, and it's also our responsibility to do so, every kid will have the chance to live up to 80 healthy years, active and contributing to Qatar and the external world. Not to mention the personal value of a healthy life together with her or his family. In order for us to maintain that medical excellence, it's crucial that we continuously challenge the way we work and how to improve our results with new knowledge perpetuously. We need to constantly improve prevention, diagnostics, treatment, and follow-up to make sure that every patient receives the most updated and efficient care. At Sidra, this is based on an integration between clinical work and research. I have led and visited several children's hospitals in Europe, but I've never seen the level of close and trusting relationship between researchers and clinicians that I meet here at Sidra. And that's also testamented by this meeting today in the more external ecosystem. The support for integration of research in every clinical decision is something I do every day here, and that's the basic of how I can support the development of new knowledge and patient outcomes. We all have to pitch in our different ways, and this is my way. When it comes to the opportunity I mentioned initially, it is as the sole provider of highly specialized pediatric care, Sidra gets to treat basically all children with serious health problems in the country. By being the sole provider, we get access to a large and complete amount of data, which is imperative in knowledge generations, as you all know. And we make use of that today, we do. But we aim to step up our game in the near future with adding agile, IT, AI, and machine learning as tools to further enhance the knowledge harvested in the interaction between clinical, research, and other data. One of Sidra's strengths is our genetics and immunology research, which is well embedded into several clinical fields, some of which will be presented today. And we have a unique and in-house and integrated setup of methods to diagnose disease from the phenotypical expression via experimental models to identify the underlying mechanisms, and eventually it will end up as possible new treatments for the patient. We have also created research programs around entire disease groups, such as diabetes, congenital heart disease, rare diseases, and others, to ensure the best possible knowledge for every kid that comes through our doors. At present, CIDRA is planning to take the next step toward effective treatments by building a clinical trial unit for kids embedded in our hospital. It's to test new medicines and also possibly medtech solutions and other treatments under safe conditions. This will not only benefit our young patients, but help researchers at CIDRA and elsewhere to test. But I also know that pharma companies all over the world will want to take use of our facilities. Currently, CIDRA is building a GMP lab to support our efforts in personalized medicine. This will enable us to administer advanced therapies, including cell and gene therapy, stem cell treatments, and support future efforts in regenerative medicine, cancer, and rare diseases. Personally, I believe that we also must bring completely new skills into medicine. For example, behavioral science, communication, art, IT, and possibly other yet unimagined competences that we need in order to succeed in our efforts to continuously improve the outcomes. Medical outcomes are powerful drivers for improvement. If you measure them and if you communicate them and use them as learning tools for improvement and creativity, they can drive innovation, and they definitely can drive passion among all staff. And good medical outcomes are, of course, crucial for the individual patient and his family, but it's also why the patients come to CIDRA for treatment, despite the fact that there are lots of good alternative care providers all over the world available for the patients. But medical outcomes have an even greater significance for the whole population of children and for society. When resources are allocated to population health, they're always a good deal for any society, and Qatar is no different. CIDRA has reached far. We conduct high-quality, internationally acclaimed research, not least demonstrated through many publications in high-impact journals year in and year out. We provide healthcare with phenomenal results in several areas, but we must understand that CIDRA, after only 3.5 years of operations, is just a startup company. We're growing, and today we produce more healthcare than ever before, but we need the resources enabled to plan ahead and continue growing in a systematic and efficient way to ensure long-term growth and excellence in outcomes for every kid. All the gains we deliver are long-term and of value to the individual child as well as society. The support CIDRA needs in terms of resources must match that to ensure that the fruits of our efforts can be harvested in a reliable fashion. This requires something from all of us, and that is that we never lose sight of the goal. And the goal is every kid counts. Thank you.
Thank you indeed, Dr. Frieden. That was excellent. Thank you for the inspirational talk. It's an absolute pleasure to have you here, and I know that you've probably been in Qatar for very long. So thank you for your time today. Next, we'll move on to our scientific. Thank you. Next, we'll move on to our scientific speakers. Just to let you know that we've selected but a few of the QNRF funded projects in pediatric research and conditions, and this is essentially a representation and acknowledgement of our support of the work in this field. With that, I don't know if you can see my slide. Right. Can you see the screen with Dr. Khalid Hussain? Hello?
Yes, we can.
Thank you.
Can you see mine?
Yeah, so before I make a start, allow me. So our first speaker is Dr. Khalid Hussain, who is the Division Chief of Pediatric Endocrinology and Program Director for Research for the Children's Clinical Management Group. He's responsible for establishing and developing the Clinical Endocrine and Diabetes Service. He leads a multidisciplinary team providing care for children and all endocrine conditions, including diabetes. Prior to joining CIDRA, Dr. Hussain was at the Great Ormond Street Hospital's Children's Charity, Professor of Pediatric Metabolic Endocrinology at the Institute of Child Health, University College London, UK. Dr. Hussain qualified in medicine from Glasgow University in Scotland, UK, and then trained in neonatology, metabolic medicine, and pediatric endocrinology in UK and Australia. At CIDRA Medicine, Dr. Hussain's research focuses on understanding the epidemiology, biochemical, and molecular mechanisms of childhood diabetes. Dr. Hussain is an internationally recognized expert in the field of childhood hypoglycemia and rare types of diabetes mellitus. He has published extensively in the field of glucose physiology and more than 300 peer-reviewed manuscripts. Thank you indeed for joining us, Dr. Hussain. The floor is yours.
Can you all hear me?
Yes, we can.
I'm trying to share my screen.
You had previously, and we had seen it well. Yes.
Can you see it now?
Yes, we can.
Can you see the screen?
Yes, we can. Yes.
I want to thank
Sheikh Abdullah, Walid, can you confirm?
Yes, Doctor, is working. As long as you see it, it means it's working.
Yes,
Okay.
it's fine.
Thank you very much. I want to firstly thank QNRF for allowing me to showcase some of the work we've been doing in the field of childhood diabetes. So thank you for the grant, and this has really helped us understanding the field of childhood diabetes in Qatar. So what I'm going to do over the next 15 minutes is give you an overview of some of the work we've been doing for the last 2.5 years in the field of childhood diabetes. I will firstly give you an overview of the different types of diabetes that occur in children. I will outline the research methodology. I will show some results and summarize the most important impact of the research. I'll show you the publications that we've already published and some of the things that I impress. I'll highlight capacity building and training and show you some future directions where I think we should be going. One of the key messages for people to take home from this presentation is that childhood diabetes comes in different forms and sizes. Children can be born with diabetes, so this is neonatal diabetes that starts from birth to 6 months of age. Type 1 diabetes is the most common form of childhood diabetes that occurs all around the world. Type 2 diabetes was rare, but now because of obesity, more and more children are being diagnosed with this type of diabetes. One special form of diabetes called MODY, or maturity onset diabetes of the young, must be diagnosed quite early because in some of these cases of MODY, you don't need to give insulin injections, you can give oral medications. And I will show you examples of this in the talk. Then there are other rare forms. There are lots of syndromes that cause diabetes. There are mitochondrial forms that are rare autoimmune. And there are some cases where we don't know the underlying etiology. So we embarked on a project about 2.5 years ago to try and understand the cause of diabetes in every child. And the aim was to stratify children into the different types of diabetes so we can personalize treatment. And this is just an overview of the project. So every child attending CIDRA diabetes clinics between 0 to 18 years was recruited into our study. We collected all the clinical and biochemical data. We measured the antibodies which will allow us to determine whether this is type 1 diabetes or something else. In some patients, we undertook detailed genetic, biochemical, and molecular testing depending on what type of diabetes we thought the child had. So what we have done, we have now classified each child into what type of diabetes they have at CIDRA. And I will just summarize in this slide, just show you. So just a few weeks ago, we had 1,403 children that were identified attending Sidra clinics for diabetes. We have now stratified each of these children into the different types of diabetes I just showed you, and these are the numbers. So you can see that type 1 diabetes is the most common diabetes that we see. Type 2 diabetes was rare until recently, but because of the obesity epidemic, more children are developing type 2. Double diabetes is a combination of type 1 and type 2. And this is a rare type of diabetes, but we've got 11 children with neonatal diabetes. So we know the cause of diabetes in every child in Qatar that attends Sidra clinics. And this is just a summary of what I've just explained. So this pie chart just summarizes all the different forms of diabetes that we see in Sidra. I'm going to highlight to you some of the most important aspects of the recent studies we've published. So firstly, focusing on neonatal diabetes mellitus. Qatar has the second highest incidence of neonatal diabetes mellitus. This is a relatively rare condition. The highest incidence is in Saudi Arabia, but Qatar has the second highest incidence. The causes of neonatal diabetes in Qatar are very different to other parts of the world. We have children where they're born with no pancreas, so pancreatic hyperplasia. So very different type of etiology here. For type 1 diabetes, Qatar has the fourth highest incidence of type 1 diabetes in the world. This follows countries like Finland, Sweden, and Norway. So an extremely serious condition here in Qatar. And when we analyze the ethnicity of children, we notice that the Qatari children are the highest numbers in terms of cases of type 1 diabetes. But here you can see we have all the other ethnicities here as well. For type 2 diabetes, again, this is very interesting. Qatar is one of the highest incidences in the GCC and also around the world for type 2 diabetes. And this is all associated with obesity. And I'll show you some of the results of this. So the age of onset, I mean, this was thought to be an adult disease, but we've got one child who at the age of 7 years developed type 2 diabetes. This was fascinating. When we analyzed the data, we noticed that 60% of the children, they had a maternal history of gestational diabetes mellitus. And as I showed you, virtually all the children had obesity. All of them had signs of increased obesity. And another very interesting, we came across a positive family history of obesity was present in either one or the other parent. And nearly every child we examined in the clinic had this sign. This is called acanthosis nigricans. And this is a marker of insulin resistance in the body. And again, when we looked at the ethnicity, you can see the Qatari children were the most in terms of numbers of the children with type 2 diabetes. So again, the key point is type 1 and type 2 diabetes are very serious conditions. And I think type 2 diabetes will become even more serious unless we don't tackle the obesity epidemic, because these children will become adults with type 2 diabetes. There's now data to show that type 2 diabetes leads early to early damage in terms of organ damage compared to type 1 diabetes. So it's a very serious condition. Now I just want to show you next few slides what impact we've made in terms of the research. So this is a patient that was on insulin treatment. But we knew that the diagnosis was not type 1 diabetes, so we undertook molecular testing, and we found that MODY type of diabetes. And here, this patient had this MODY type, and in these cases, you can stop insulin injections, and you can start oral medication. So the child was able to stop insulin injections. This is a fascinating family where 4 children had diabetes. And again, our molecular testing showed that they have a type of diabetes that does not need treatment. These patients were on insulin injections. So you can imagine if you're a parent, I come along and tell you you do not need insulin injections. That will really make your day. Similar here, another type of MODY. Here, no treatment was required again because the MODY was due to this gene here. Again, another example, again, another type of MODY where, again, we were able to stop insulin injections and start oral medication. Similar here. So these are just giving you an examples of the type of translational impact that we can have if we know the molecular biology. And the most important patient we've recently had is this patient. This is a patient who developed diabetes at the age of 4 months, very, very early on. Any child that develops diabetes before 6 months normally has what we call neonatal diabetes. So we knew that this child did not have type 1 diabetes. Oops. So we undertook, the child was originally on 4 injections of insulin a day. So you can imagine a 4-month-old baby having 4 injections a day. We undertook molecular testing in Sidra, and within 2 weeks we had the result. And we had a molecular basis for the diabetes. And we knew that we could treat this with oral medications. So this patient was, the insulin injections were stopped, and we pushed the patient onto oral medication. And the evidence base for this is shown here. So these parents are extremely delighted. No more injections for this patient. So beautiful example of the translational impact that we can have. Everything was done at CIDRA within 2 weeks. This would have taken months and months and months previously if we did not have the facilities. This is what we've published so far. So all these manuscripts have now been published, and this is from the grant we've got just had. And this was just published at the weekend. So this really sets the scene for type 1 diabetes. In this manuscript, we describe the clinical features, the epidemiology, all the antibodies, and the molecular basis of type 1 in children. And these are the manuscripts that we're working on at the moment. Some are in press and some under review, and I'm hoping that by the end of this year, I hope all these manuscripts will be published. So we'll have quite a lot of information on childhood diabetes by the end of the year. In terms of capacity building, I have four PhD students who are working on diabetes. So this is Sana, she works on a rare type of diabetes called Macroenvironment. This is Aman, she works on another rare type of diabetes. And Basma is my research fellow. She collects all the information on the information that I've showed you on type 1 and type 2 and all the other rare types of diabetes. And Idris does some work on obesity and some work on diabetes. And Kulthum is working on short stature. So in terms of future direction, we really want to now push this forward. We want to now start practicing personalized medicine because we know what type of diabetes every child has. And we want to be ready for the next phase of new therapies which will come along. And this is a complex slide, but I just want to show you. So this is a patient with type 1 diabetes, and this is what's coming along in terms of future therapy. Type 1 diabetes is a condition which at the moment is managed with insulin injections or insulin pumps, but we need to be able to be ready for the next phase of therapies. And there's a huge amount of work in trying to suppress the immune response, targeted immunization, targeted immunomodulation, and also curative therapies in the form of stem cell therapy. So we should know which child has type 1 diabetes so we can proceed with precision medicine. And type 2 diabetes at the moment, we give metformin, which it doesn't really work in most patients, or we try and ask them to lose weight, again, which is a very difficult thing to do. But these are the sort of drugs. So this is a GLP-1 receptor agonist coming along for the treatment of type 2 diabetes in children. So this is precision medicine for type 2. And this is my sort of vision. This is where I want to go. I want us to become an internationally recognized center for the diagnosis and treatment of all forms of diabetes in the future. We want to be the center for testing for these types of diabetes that I showed you and have a center for new treatments for all the rare forms of diabetes. I want to acknowledge the support of a large number of people that I work with. These range from my own lab, from the physicians, patient educators, nursing staff. Basically, there are so many people that I work with in the hospital and from the research division. And I really need to acknowledge the support and help of everybody. And I want to thank QNRF once again for helping me with this work. Thank you so much.
Thank you very much indeed, Dr. Hussain, for that work and for your excellent presentation. I'm not entirely sure whether you have time to stay with us until for the very end for questions, or if not, I can just maybe pose 1 or 2 questions here.
I've got a clinic to go to, so I won't be able to stay.
I just want to say it's a truly colossal task that you've taken on, and it's really excellent work. And I'm truly looking forward to the end and to see what are the outcomes. 1 question before you go, at least I have several questions, but I'll just pose one since I know you need to be away. 1 question that has come in is, from your current findings on the high prevalence of type 1 diabetes in Qatar and as the 4th highest incidence prevalence in the world, what is the best strategy you think should be taken to lower that?
Well, the problem with type 1 is we don't know what actually causes type 1. We don't know what the trigger for type 1 is. And I think until we don't find the trigger, it's very, very difficult to sort of prevent it or stop it. And this is exactly what's happening around the world as well in some of the other countries. For example, in Finland, a very high incidence. And there is research in trying to understand what is the actual trigger, what is the actual underlying mechanism, what is the thing that starts damaging the pancreas. So unless we don't know the mechanism at the moment, we can't prevent it, we can't stop it. So the research is focusing on trying to look at the trigger for type 1 diabetes.
A very quick question before you go, a second one, is you had mentioned somewhere, I'm not entirely sure whether you, maybe I missed it in your talk, that there's a possibility that you may not be able to identify children who have type 1 diabetes, or maybe those who are not classified as type 1 or type 2. And you hope to place these children into a gene discovery research program. Can you just sort of enlighten us where you are with regard to that? i.e. you had envisaged sort of genomics and metabolomics studies on that. Where do you stand currently on that?
So we've got one group of children that are labeled as type 1, but we cannot find the antibodies. There are about 52 of these children. So that's a group we're quite interested in because we don't know what the actual cause of the diabetes. It could be other antibodies that we don't know about, but we're really fascinated in this group. And I've got people in CIDRA here that are interested, and I'm working with those people doing more metabolomics, proteomics, and genomic studies in these patients. And then there are other patients where the cause of diabetes is completely unknown. Again, we've got a few patients, not that many, but because we managed to find the cause in most, there are 3 or 4 patients where we don't understand the cause. And again, we're doing a lot of genomic work on those.
Okay, with that, thank you so much indeed for your time. With that, we'll leave it and wish you the very best and every success in this project. We're truly behind you. Thank you so much for this colossal work. Thank you very much.
Thank you so much.
Thank you indeed. Thank you. I don't know if if my slides are visible to you.
Not yet.
Okay, let me see if I will try to get. I'm trying to get the picture of Dr. Sahela.
They are now. Yeah, now we can see them. Thank you.
Okay, thank you so much. So for our second speaker, we have Dr. Sahela Al-Khadour, who is the Director of Maternal and Child Health Program at CIDRA Medicine, Qatar. Dr. Al-Khadour received her PhD in microbiology and immunology from the University of Louisville in Louisville, Kentucky, I think that is, USA. And before joining CIDRA, Dr. Al-Khadour worked as a postdoctoral fellow in the Signal System Unit Laboratory of System Biology at the National Institute of Allergy and Infection Diseases at the National Institute of Health, the NIH, in Maryland, USA. Dr. Al-Khadour's laboratory in CIDRA focuses on characterizing the role of microbiome in health and disease, with a particular emphasis on maternal and child health. In her project, Dr. Al-Khadour uses integration of metagenomics with other omics, aiming to identify pediatric predictive markers for various diseases and pathological conditions. Dr. Al-Khadour is an adjunct assistant professor at the College of Health and Life Sciences and Hamad bin Khalifa University, and an adjunct assistant professor at the Department of Biomedical Sciences, College of Health Sciences in Qatar University, and has over 45 peer-reviewed publications. Dr. Al-Khadour, it's a pleasure to have you here. It's over to you for your presentation. Thank you.
Thank you, Dr. Fatma, and good morning to everyone. Thanks for the organizer to allow me to present my updates on the QNRF project and PRP10. I'm trying to share my screen. Let me know if you can see my slides.
We can see it now, Dr.
All right, thanks. So today I'll be given an update on the project titled Towards a Systems Level Understanding of Pediatric IBD in Qatar, Interplay Between the Immunogenome and the Microbiome. This is an NPRP 10 funded grant. I'll be going through the background, the design of our studies, the methods that we use, the current progress and some results, our future work, how can our result be translated to the patient, some outcomes and acknowledgements. So before I go in the details, I prefer to introduce the topic about IBD, or inflammatory bowel disease. It's characterized by chronic immune-mediated inflammation of the gastrointestinal tract. It is usually a progressive disease with a destructive course. There is 2 major subtypes of the disease, Crohn's disease and ulcerative colitis, but there is a 3rd subtype that is kind of unclassified, because even after assessment by the clinical team, the clinicians will remain unsure to classify it either as Crohn's disease or ulcerative colitis, hence its name as IBD unclassified. It has a high incidence in the US and in Qatar as well. The disease is multifactorial. with genetic susceptibility, environmental triggers, luminal microbial antigens, immune response, they all play a role in the onset of the disease and its progression. If we think of the disease course, it goes from a flare or a relapse into a remission, then into a flare again. We don't know what usually triggers the relapse or the remission after treatment as well, but we tried in this project to understand how can the relapse and remission progress in a given patient. The gold standard for diagnosing IBD is usually colonoscopy, endoscopy, imaging, a lot of invasive techniques, and assessing the severity of the disease. There's a lot of work that is done on the clinical side to understand the level of the disease, and there is a strong need to develop non-invasive technologies and try to identify disease biomarkers that we can help the clinical team to diagnose the disease and predict its course, whether a patient in remission is going to progress into a relapse and when. So with that in mind, we designed the project back in 2017 to have the following objective. First, because when we assess IBD in Qatar, there was no clear database of how many patients have the disease or where the progressing, if they're IBD, if they're Crohn's or IBDU or ulcerative colitis. So we decided to build our own database for the IBD kids living in Qatar. And we wanted to do in-depth molecular profiling for the pediatric IBD, and then later do some data integration to look for biomarkers that can help us predict the disease onset and its progression. So this was the simple design of the study. We wanted to recruit 85 patients with either Crohn's disease or ulcerative colitis, and matching healthy controls. These will be coming to the Sidra IBD clinic. We follow them up from the first visit, and then every 3 months after that. If the patient felt any unwell episode, they will be also recruited in the study. As I mentioned, the disease goes into a well and flare episode, so we aim to also capture the different time points of the disease. We finished the sample collection, but we continue the biorepository as we speak. We collect a lot of data from the patient, but we also collect saliva samples, stool sample, and blood sample. The saliva and stool sample will go for 16S metagenomics. I'll explain that in the next slide. And then the blood will go for assessing the immune response. So when we speak metagenomics, we refer to microbiome, and this is the study of what type of microbes are living in a given site in our body. Our body is full of microbes from the inside and the outside. These microbes are good microbes when they are maintained in balance, so they're called commensal bacteria. When there is any imbalance or addition to pathogenic bacteria, this creates the imbalance / dysbiosis, and this can trigger or contribute to different diseases, including diabetes, including IBD, including asthma, and so on. The list can go for a long time. The majority, the biggest composition of the microbiome in our body would be in the gut or the gastrointestinal tract, and this is composed of 2 major families, Firmicutes and Bacteroidetes. When we ask the question, what is the microbiome in the patients with IBD? Majority of the data present in the literature is, of course, done on the adult population, and very few are done on a pediatric population. We try to ask whether from reviewing the literature, we can find a signature that can tell me this patient is a CD, or this patient is a UC, or this is a healthy control. Unfortunately, the current data doesn't indicate that there is a clear microbial signature that can differentiate between the two patients. We can only say that the IBD microbiome is different from a healthy microbiome. There are few microbes that are up and down in either subcategory, but there is no clear indication on how is that changing, especially in our pediatric population or kids with IBD. So we published our assessment to the literature in the first paper coming out of the grant, but we also went deeper to understand how the microbiome and the immune system are interacting in the patient in IBD. So as I mentioned, there is a genetic susceptibility. There are environmental factors such as the diet that these kids eat, and then there is microbial dysbiosis. They all play a role in promoting or predisposing kids to IBD. There is, of course, some loss of integrity in the epithelial lining that would allow those microbes to translocate, and hence they will be in a continuous interplay, stimulating our immune cells and causing this excessive stimulation. Again, by reviewing the current literature based on the adult population, we were able to define preliminary signatures of immune phenotypes for Crohn's disease or ulcerative colitis, but we wanted to dig deeper and understand how is that changing in our pediatric cohort. This is again another paper that was based on our assessment to the literature prior to working with our own patients. In parallel, there was a lot of work being done in the lab. We wanted to start from scratch and build our own panel. This is a real photo from my lab with the thousands of strips that were prepared with the ligands that we wanted to stimulate the immune cells of the blood collected from the IBD patients and the control in order to assess how their immune system is reacting when they are in a flare or in a remission condition compared to the healthy controls. So before we started with the patient, we built the database, and this is thanks to the bioinformatics team here at CIDRA. We went through different series of or versions of the database until we were satisfied with a database that captures all the information we want about our patient. So once the patient is coming to the IBD clinic, we can send them, if they're happy to be part of the study, they immediately enter in this database where we collect a lot of information in the registration form and their baseline visit, what they eat, their demographic, socioeconomic information, clinical information, phenotypic information. Everything is captured in this database, and of course we follow up the patient. If the patient gets hospitalized, there will be a hospitalization activation form, and all the track of the patient since they entered the study will be captured in this database. So what is our current update? We have successfully recruited 85 IBD patients from the clinic. We are following them up. We are now in visit 6. This is reflecting the number of samples that we collected for blood, saliva, and stool. We have recruited 57 controls. So these are healthy controls. They do not have chronic diseases, and they're age matching our IBD patient. As I mentioned in the baseline, there is a baseline questionnaire, disease index questionnaire, what medication they take, their history, and then the questionnaire will change based on the follow-up visit. We have about 340 samples that went for metagenomic sequencing for saliva, 240 that went for 16S for the stool. These are for assessing the microbial composition of the sample, and about 1,100 samples went for blood transcriptomics. So this was a huge work done in the background to prepare all the samples before they go for these 3 pipelines. So to just briefly explain how do we deal with the saliva and stool samples to assess the microbial composition. As I mentioned, we have the patient, we have the healthy control, we collect the samples. The stool or saliva will go first step to extract the DNA, then amplify the 16S ribosomal RNA gene. This is a gene that is present in all the bacteria. So we amplify it, we assess the quality, the quantity, we prepare libraries, and then we do sequencing. It's very similar to the human genome sequencing, but this is only microbial sequencing to tell us after assessment what type of microbes were living in this community, in this sample for a patient or a control. We do the same for stool, obviously. So this is our preliminary results based on the samples that the 200 samples that we collected so far. There is already a clear indication that we have differences between the saliva sample of a control or the saliva from a Crohn's disease patient or a saliva sample from an ulcerative colitis patient. This is all significant, and here we include the IBDU, which is the subtype that is not classified. When we look at the alpha diversity, this is the diversity significance within the sample itself, and this is beta diversity. Again, we have significant difference between CD versus control and UC versus control, indicating that the saliva sample of these patients is already looking different from those of a healthy control. To be more specific, we looked at the differences at genus level, and we see that there is specific bacteria that are different between patients with Crohn's or ulcerative colitis or IBDU, and these are mainly Neisseria, Provotella, and Streptococcus. Just to make it closer to your mind, Streptococcus is very well known to cause the dental cavities in the kids, so it's of importance, is known to be a pathogenic bacteria. We did the same for the stool sample. Again, there is differences seen in the beta diversity, which is difference between samples, significant values in the composition of the stool sample collected from Crohn's disease or controls or IBDU, indicating again and reassuring us based on the data from the adult, there are differences known to be there causing dysbiosis in patients with Crohn's disease or ulcerative colitis compared to healthy controls. When we zoom in, we found that the genus Faecalibacterium, Enterobacteriaceae, Neisseria are also different between these groups. Next, when we moved to the second area we wanted to look at was the immune response. So we designed our own internal pipeline to look at the blood transcriptome and proteome. So when we collect the blood from those samples, if you recall the plates that I showed you in the photo, this will be one of the plates. We have ligands that are already embedded in these plates. So we bring the blood, we mix it with the solution, then we add it to the wells, we incubate it 6 hours, and then we lyse the cell, or we take the supernatant. So we look at the secreted proteins, we look at the transcripts to assess what type of genes are up or down regulated in the patients versus control in response to 16 different stimulations. So this is really a very in-depth understanding of how the immune system of these patients is responding to specific stimuli. And here is the preliminary data. I can start from this side. This is data from one patient. The patient has...
You have 2 minutes left. Can you please wind down? Thank you so much.
Sure. This is just to reflect how the patient, same blood sample from the same patient at different time points of the disease, moderate, severe, or remission, is already clustering differently. So it was also indicative of that the expected hypothesis, that the planned hypothesis that we had. So where we are right now with the project, we are in year 4 in the 3rd quarter. We are wrapping up the project in February. We're right now in the data analysis of the microbiome and the immunogenome for integration. We have contributed to national capacity building with the local recruitment of 2 ladies, Marwa and Dua, who are working on the project as a research coordinator and research specialist. We've trained 3 students with the advanced omics technologies that we have, 1 master student from HBKU, another undergrad medical student, and 1 BS student from Ontario, Canada. We know after discussion with the clinician that this project has a great value for translation onto the clinical side. It's a promising use of saliva sample instead of stool sample because these are less invasive and easy to collect. We can identify microbial signature that can help identify the patients with CD or Crohn's compared to healthy control without the use of invasive technologies like colonoscopy or endoscopy. And then we hope that by merging or integrating the microbial and immune signature, we can help finding precise clinical decisions for our IBD. We've published 2 papers. One is currently under process. This is all outcome from this grant. I want to acknowledge my team, specifically Dr. Mathieu Garon for his input on the immunology side, the RedCap database here at Sidra, Pediatrics Department, Dr. Mohammed Handous Rahal for recruiting the healthy controls for us. Definitely the IBD clinical team, Dr. Ma'moon, Dr. Tony Parag, Dr. Fatma, Ms. Lin, and Mr. Nazira, the nurse who's coordinating all these efforts, and QNRF for funding my project. Happy to take questions if there is time. Thank you so much.
Thank you very much indeed, Dr. Sahela. If you don't mind, due to time restraints, we'll try to take some questions right towards the end, so please stay with us. With that, we'll move on to the next speaker. Are you able to see my screen?
Not yet.
Is that visible to you?
No.
Not yet.
Right, bizarre. Let's start again. For you. I'll try one more time. Are you able to see that?
No.
If not, I'll move. Okay, so we'll move on. So for our third speaker, we have Dr. Kiara Cuna, who is the director of the Advanced Cell Therapy Core and attending physician in pediatric oncology and hematology at Cedar Medicine. She also covers the role of acting director of Human Subject Research Protection Program since January 2019. She has 20 years experience as a medical doctor with board certification in pediatrics and pediatric hematology and oncology at the University of Pavia, Italy, and has a master's in pediatric hematology. In Italy, she was directly involved in the hematopoietic stem cell transplantations activity at the renowned pediatric hematology oncology unit, which had performed more than 2,000 HSCTs. At CIDRA, she has been working on the development of the Advanced Cell Therapy Core, including a cellular therapy unit for the delivery of cellular products for tissue, cell and gene therapy, and research projects on mesenchymal stromal cells and pediatric leukemia. Her activities currently range from hematopoietic stem cell processing as part of the bone marrow transplantation clinical program to several projects of cell therapy, gene therapy, and regenerative medicine. She has additional research interests in pediatric leukemia, inherited bone marrow failure syndromes, graft versus host disease. Dr. Cuna is author of more than 20 articles published in peer-reviewed journals. So with that, I'll hand it over to you, Dr. Cuna. The floor is yours. Thank you.
Good morning to everybody. I guess that you can see my slides.
Yes.
Yes. So good morning again, and thanks to the organizer, Dr. Fatma, for inviting me to give you an update on the results of the NPRP8 grant that is entitled Microenvironmental Targets for Restoring Anti-Tumor Immunity in Childhood Acute Leukemia. So first of all, the background, childhood acute myeloid leukemia is a rare and heterogeneous disease. And nowadays, the chemotherapy remains the standard of care, despite the recent approval of other drugs, novel drugs. And this can, with the chemotherapy, the survival rate is approximately 70%. Approximately 5% of kids with AML has refractory disease and 30% experience relapse. And overall, for these two categories, the prognosis is extremely poor. So there is the need to develop novel treatment strategies, including also targeted therapies. Let's start from the microenvironment, since the grant was mainly focusing on the microenvironment. And when we talk about tumor microenvironment, we talk about the milieu where the tumor cells are living together with stromal cells and also with immune cells. And you see in this cartoon that the immune cells can be activating immune cells like CD8 cells, NK cells, dendritic cells, but also immune suppressive cells, mainly T regulatory cells and macrophages, so myeloid-derived suppressive cells. And if we go specifically to the AML environment, microenvironment, this is extremely suppressive for immune effector cells, meaning that the immune response against the blast is impaired, and this can be due to direct contact of the leukemic blast directly with the cytotoxic cells or through the secretion of several cytokines or molecules. And our initial plan was mostly focused on IDO molecule. But also we know nowadays that other cells are contributing to help the tumor cells to evade the immune surveillance, and these cells are T regulatory cells. This is the paper that was written by Sergio Rutella in 2014, and it was the background of the grant application. He published that patients with IDO expression, high IDO expression in the tumor had an event-free survival and overall survival significantly worse than other patients. So there was the hypothesis behind the grant that we have two subsets of AML patients. One so-called inflamed AML with the infiltration of T lymphocytes able to induce through the interferon IDO expression. And in this case, we could eventually use IDO inhibitors or other checkpoint inhibitors. And another set of patients with no inflammation, no immune response in AML, where we have to think about additional strategies such as antigen-specific T cells or CAR T cells. I can tell you now, this was done in 2014, that several trials on IDO inhibitors failed to visualize any benefit for the patient. And so let's come to the work that we have done specifically at CIDRA in collaboration with Sergio Rutella, that now is working in Nottingham Trent University, and with our collaborating institution in Karachi, Pakistan. We have recruited 9 AML patients at CIDRA, 14 AML patients at Aga Khan University, and to enlarge the cohort, we additionally have other 19 AML patients from my previous institution in Italy. They are all pediatric patients. Since we were mostly interested in the macroenvironment, we applied a platform that is available at CIDRA that is called NanoString, and specifically a panel, pan-cancer IO360, that is a panel of 770 genes that are involved in the interplay between the tumor, the microenvironment, and the immune cells. So this is a panel that is extremely interesting, especially for understanding the impact of immunotherapies and also to interrogate the immune evasion. So our cohort was mainly consisting of 35 pediatric AML at diagnosis. We excluded the relapse patient. 3 samples failed the quality control and were excluded, and so and also other 7 samples with unclear clinical information were excluded. So we come up with 25 patients that we categorize in 2 cohorts that we defined sustained complete remission and non-sustained complete remission. Sustained complete remission being defined as the response and sustained remission after only the first line of treatment of chemotherapy. While in the other category, we include all the patients that failed the first line treatment, that went to bone marrow transplantation or passed away due to the progression of the disease. And this is our results with an analysis called the single sample gene set enrichment analysis, mostly focused on the immune system, so on the interferon module, and on the immunological constant of rejection. That is a panel of 20 genes involved in the immunological response, and dealing with the T helper Th1 lymphocytes, the CD8 lymphocytes, immune effector factors like granzyme, perforin, and granulosin, other chemokine ligands, and immunosuppressive regulatory genes. And you see that it's quite, the results that we had are quite striking, because you have here in purple, this is the cohort of patients that maintained a complete remission over time with only 1 line of treatment. And you see that there is an overexpression, a clear overexpression of all the modules related to the interferon production and to the immune response. And if we plot the 67 transcripts that are differentially expressed in the patient with sustained immune response towards the other cohort, with the Vulcano plot, we see clearly that the molecules expressed in the patient, or more expressed in the patient with worse outcome, are suggestive of a microenvironment that is highly immune suppressive. Is suggestive for monocyte, specifically for the presence of myeloid-derived suppressing cells. While on the other side, we have patients who did well with a good outcome, where the immune response was activated. So we have an interferon pathway activation and also the T cells activation. And if we plot these the genes upregulated in complete remission in a functional enrichment analysis through string, we clearly see how the pathway, the color code is here on the right, are clearly related. All these genes are related to the pathway of interferon, tumor necrosis factor, interferon alpha and beta adaptive immune cells. But we wanted to go further, since we acknowledge the importance of epigenetics, so of control of gene expression through other and additional mechanisms. We wanted to understand whether there was any impact to the gene expression coming from the methylation pattern. So 15 patients in this case were considered, and we are currently working to include also the remaining patients that are still missing in our analysis. And you see another striking results. Here again in orange, you have complete remission, yes, sustained complete remission, and the other on this other side of the heat map. This is an unsupervised hierarchical clustering, so naturally this patient cluster and in a different way compared to the other group. And if we go a little bit deeper, again we focus on these are the patients that go well, and if we consider the hypomethylated genes that are approximately 90, with and 58 are known, and we know usually there are treatment nowadays used, therapies used in AML with hypomethylating agents, because it's known that the majority of these genes are more functional in their hypomethylated form. And let's see at these 58 genes that were found more hypomethylated, so probably more functional in the complete sustained complete remission patient, and they are mostly related to they are all related to cancer. If we plot again the genes in the string functional analysis. So the conclusion for this first set is that the strong differences were identified in gene expression of patients with sustained complete remission versus non-sustained complete remission, same for the methylation pattern, and that we suggest that interferon and T cells activation in sustained complete remission, while immune suppression in non-sustained is present in our patient, with a prominent role for epigenetic mechanism like methylation. And just 1 minute for the omics approach that we are applying to all our patients with leukemia, including also the AML that we discussed till now. And this is an abstract that we submitted to and we presented to ASH, and where we showed that the TP53 is highly expressed in intermediate standard risk compared to the high risk acute lymphoblastic leukemia. No differences in AML. And looking specifically to the TP53 and the apoptosis pathway, we were wondering whether there were any mutations for the low expression of the RNA in P53 in the ALL patient, but they were not. And there was only one mutation in the AML.
You have 1.5 minutes, please, yeah. Dr. Can you please wind down? Thank you so much.
Yes. And then we looked at the methylation pattern. Again, no differences between the two groups. And strikingly, again, it was the macroRNA expression that was differentiating the high-risk leukemia against the intermediate and standard leukemia, suggesting that there is a unique program of control the TP53 in an epigenetic way in the high-risk ALL leukemia. So we are currently validating this data on a larger cohort, and we are also adding proteomics analysis. One example of clinical application of the omics approach is one patient that was treated here at CIDRA, and where we found a mutation in c-KIT, mutation that was not previously associated to AML. And but why this is important? Because if we sequence or if we study in deep all our patients, we are able to identify mutation, like in this case, that can benefit from other additional therapy, like tyrosine kinase inhibitors, like in this case. And this is a key message of why this is important, why what we are doing is important, because we understand increasingly, and it's increasingly known that for addressing the leukemia stem cells, we need to act upon different mechanisms. Not only the chemotherapy, of course, the epigenetics, as we saw, the microenvironment, immune therapies, and regulator of apoptosis. Publication are 5 publications with Sergio Rutella and other 2 that are in with the manuscript that is in preparation with the data that I showed you, and the acknowledgements with my fantastic team, Advanced Cell Therapy Core, and the Integrated Genomics Core, the cancer program led by Davide Bedognetti, and of course my colleagues in the pediatric oncology and hematology, Dr. Ayman, Dr. Naima, all the consultants, hospitalist fellow, and the nurse, and our collaborators in Italy, in Bolzano, in Italy, and in Pakistan and UK. Thank you for your attention.
Okay, thank you indeed, Dr. Cuna. Thank you so much for that. We'll need to move on, so if you bear with me, we may have some questions for you at the end. And also, yes, please sit with us for the panel discussions. We may need you there as well. So next, I'll try this one more time with regard to sharing my screens. Let's see how much I can. I don't know if you can see Yunus Matlab?
Yes.
Okay, excellent. Okay, so for our next speaker, fourth speaker, we have Dr. Yunus Makrab, who is the principal investigator and head of medical and population genomics lab at CIDRA Medicine. He joined in 2015 from Eli Lilly, where he led computational genomics research at the neurogenetics discovery unit to identify and validate drug targets and stratify patients with neuropsychiatric disorders, including schizophrenia, ALS, and Parkinson's, working closely with Psychiatric Genomics Consortium, PGC. Dr. Makrab obtained a PhD in bioinformatics from the University of Cambridge, UK, followed by a postdoctoral fellowship at the University of Oxford, 2010. Next, he moved to the pharmaceutical industry, initially Lonza Biologics, performing in silico biopharmaceutical engineering, and subsequently Eli Lilly, where he led research in early-stage drug discovery and various areas of neuropsychiatry. Upon joining CIDRA, Dr. Makrab helped establish research programs in population and medical genetics, and is co-founding member of the Qatar Genome Program Research Consortium. He currently holds academic appointments at Cornell, at Cornell Qatar, at HBKU. Dr. Makrab is a co-author of more than 45 publications. With that, I'll hand the floor over to you, Dr. Makrab. Thank you for joining us.
Thank you so much, Dr. Fatima, for the kind introduction. Assalamu alaikum, everybody. I would like to share my screen now. Can you see it, Dr. Fatima?
Not at present, Dr. Yunus.
Okay.
Try to reshare it, Doctor.
Okay.
Yes, it's now we can see.
Okay, very good. Well, thanks so much for giving me the opportunity to present today to you on this QNRF research outcome seminar to discuss and to report also the research outcomes from our QNRF grant entitled Building Haplotype Resolved Genetic Maps as a Premium Resource to Improve Diagnosis and Characterize Developmental Disorders in the Qatari population. And then if I can go on full screen mode. Yeah, okay. So neurodevelopmental disorders, or in short NDDs, affect up to 5% of children worldwide, and they have they affect primarily cognitive functions in addition to a variety of other neuromuscular and organ functions. There is a very important genetic component to NDDs, and mutations have been found in over 1,000 loci associated with this group of diseases, ranging from short single nucleotide variants, which can either lead to loss of function or gain of function, in addition to structural variants which impact thousands of nucleotides, and they can be both inherited and also de novo. The prevalence of the specific monogenic dominant forms of NDDs has been estimated to be about 1 in less than 300 individuals, which is greater than the combined impact of the well-known trisomies of 13, 18, and 21. And despite being individually rare, the group of NDDs cumulatively have a high or lead to high morbidity and mortality in the population. And economically speaking, affected children have a very complex health needs. And in fact, 30% of all children that are admitted to inpatient clinics have some sort of genetic disorders with them, and more than 50%, more than half of all acute inpatient healthcare costs is actually associated with this exact type of patients. And there isn't a definite estimate of the numbers here in Qatar, but it's likely to be higher due to the high level of consanguinity and therefore the enrichment for recessive genetic disease. Some background also about the genetic structure of the Qatari population, which is very important when we study patients, especially with genetic disease. So we recently conducted a study looking at trying to understand the genetic structure of the local population, where we tapped into a cohort of more than 6,000 whole genomes from QGP phase 1 cohort that has been recruited by Qatar Biobank, and identified 5 distinct ancestries in the local population. So these include Peninsular Arabs, and I'm pointing here with my mouse, as well as general Arabs. So these are being basically the largest components. And then we also have West Eurasian Arabs and a very small fraction of what we call African and South Asian Arabs. And then basically we have a fraction of that, which is an admixture of either of these ancestries. And as you can see here in the PCA plots, they form distinct clusters in space when you compare them to other populations. What is really interesting is that these ancestries are actually quite relevant in other populations from the wider region of the Middle East and also the world. So they're actually, these ancestries that we've discovered in the local population are actually generalizable to the wider Arab and Middle Eastern region. What this means is that we have an opportunity now to be able to apply population level stratification of individual genomes in order to be able to identify the relevant background genetic information matched by population, and therefore be able to more precisely and more accurately identify rare disease variants. At Sidra Medicine, so we have a broad strategy for trying to enhance the diagnosis and also the characterization of neurodevelopmental disorders, and we tap on quite a few pillars. What is first quite important is the disease cohort. So we're working very actively with our clinical colleagues, both at Sidra and also Hamad, to recruit families with a wide range of neurodevelopmental disorders. And also we're gaining access to a lot of cohorts, either from the local population, i.e., for example, Qatar Genome Program and QBB, but also multi-ethnic data sets from across the world, which is very important when we try to look more comprehensively into the genetic burden of MDDs. And what we're doing is we're basically building disease cohorts around this specific area, and we're involving here not only the collection of blood samples, but also other surgical samples, since many of the patients undergo neurosurgical procedures. And we apply state-of-the-art genomic characterization techniques to try and understand not only patients, but also their parents and their siblings, etc. And we also are engaging in massive efforts to try and also deep phenotype the patients by not only looking at measurable phenotypes, for example, from neurology or pathology or imaging, but also psychiatry, and trying to more identify better ways of stratifying and also diagnosing patients. And of course, we're very lucky to have access to some of the state-of-the-art platforms in terms of HPC and computational platforms to be able to apply data integration analysis. So now talking specifically about the current NPRP project, funded by QNRF. So our main mission there is to build a data resource using state-of-the-art sequencing technologies in order to bring a better diagnosis and more understanding to what's happened at the molecular level for developmental disorders in Qatar. So our main objectives are as follows. First of all, build a family-based disease cohorts recruited from the local population here in Qatar, and this involves recruiting the subjects, phenotyping them, and also collecting their blood samples and other related tissues. Construct de novo reference quality genome assemblies for a few individuals that represent the Qatari population genetic ancestries, using long-read sequencing and also the latest genome mapping technologies from PacBio, BioNano, in addition to the more conventional techniques of Illumina short-read sequencing. Characterize the spectrum of short-read or short variation in addition to complex variation in the study participants, and prioritize candidate disease variants in the probands, and with the focus on looking at about 9 trios with the highest resolution that we can do with those. And the outcome basically at the end also kind of takes us nicely towards building the study portal that facilitates the clinical interpretation of any future variants that would affect families of similar phenotypes, and also to allow us to be able to disseminate the results from this study to the wider community. So in terms of results, first of all, we've been able to start building the patient cohort for this study. So as basically a number for guidance, from 2016 to up until the end of 2020, more than 5,000 patients have had primary appointments in various relevant clinical departments here at Sidra. About 30% of them were Qatari nationals, and the rest of it, which is the majority, coming from mainly countries from the Middle East and Northern African region. We managed to recruit blood samples for about 45 families with unresolved cases. That's a total of 250 subjects. And we also profiled genetic ancestry ascertainment for the samples and their for the subjects and their parents and siblings, etc. And we basically, as we show here, they show a spread across the various main ancestral groups that we've identified in Qatari population. So we've also tapped into their electronic health records that have been populated within the Cerner system at CIDRA, and we've been able to systematically map what would have been descriptions of the various symptoms or clinical manifestations into formal HPO ontology. So this is basically a formal HPO terms that allow a more systematic analysis and aggregation of the data. Another very important progress that we've been able to achieve is that we've generated short-read sequencing data for about 206 samples from about 32 families, in addition to PacBio and BioNano. These are high-resolution techniques for about 22 subjects. We've established state-of-the-art bioinformatic processing pipelines, which also include quality control, which is very important, especially for these cutting-edge technologies where methods are still yet not mature at the level of some of the conventional ones, for example, short-read sequencing. And also, we managed to produce high-quality sets of single nucleotide polymorphisms as well as indels for about 166 samples, as well as having called structure variants from PacBio and BioNano data for quite a few samples from those as well. And what's also a very important progress I would like to report here is the generation of high-quality draft de novo genome references for about 4 subjects from 3 reference families. And this is just some metrics to tell you about the contiguity of these genomes in terms of scaffold N50. So you can see here that by applying assemblies based on PacBio and then doing hybrid scaffolding using BioNano, we've been able to stretch the N50 to the range of up to 70 megabase, which is greater than state-of-the-art GLC38 genome reference. In terms of gaps, we're still working on that because still GLC38 is state-of-the-art in terms of the various manual techniques that have been used in order to close gaps, whereas essentially we're still hitting very good numbers here in terms of like 14%, 15% for some of the samples, and also gaps as low as 1% for some of the families. So that's really quite encouraging. Another result that we would like to talk about is our implementation of Congenica platform for doing family genetic analysis, and this is based on state-of-the-art platform that has been widely adopted. So started off in the UK, but then became very rapidly adopted across various research hospitals across the world, and also notably by Genomics England. So we've been able to deploy their pipelines and framework in-house at CIDRA, and this allowed us to start uploading our families, analyzing our families, and creating dashboards for them, and also capturing a variety of details, including pedigrees, and also various annotations on the subjects themselves, the relationships, and providing at the end reports that can be shared with clinicians in order to further deliberate on the actual outcomes in terms of genetic diagnosis for these families. So this has been incredibly challenging because a lot of these families, essentially, we came up that we've been looking at, essentially are prioritized for those that are undiagnosed. So meaning there has been some basic genetic testing that's been done at CIDRA Medicine, and none of them returned any results on that. So we kind of dived into those and identified up to 20 genes that we think, and this is just an example of one of the families, so that we think are actually plausible genes that contain some variants that are likely to be pathogenic variants. But right now we're still working in terms of increasing our confidence about this discovery. So we're looking at really, going back to the QC stages of making sure that those variants are real, and also tapping into other data sets
Can I please ask you to wind down, please? You're running towards the end of it. We've got just
Yeah,
over a minute left.
Sure, definitely. So basically, currently we're just tapping into some of those international data sets that would allow us to, from genomic SyncNet, from Synapse and NDD, from DDD from the UK, that allows us to look at multiple occurrences, possibly for these variants. So just to wrap up by essentially summarizing the main outcomes, to say that we've managed to build a database for this is database for NDDs. Including highly curated phenotypes and genomic data. We're currently working on 2 manuscripts on specific family cases, as well as to report on our work on de novo references building. And then finally, we've developed, we think, some real capacity at CIDRA that could serve both Qatar and the region in terms of being able to apply long-read sequencing to look at undiagnosed genetic diseases. And this also has been done with generous support from CIDRA in-kind funding, and we've been able to also train few graduates and data scientists as well on this project. I would like to acknowledge the various collaborators and also the funders, QNRF, obviously, CIDRA internal funding, and also primarily would like to really give a big thanks to the great team that we have in CIDRA that's been working on this project, namely Rozaimi Rozali, Mersad, Haroun, Ali, as well as Radi, Rima, Sara, and Shabir, as well as our key collaborators here, Khalid Fakhro and also Fatima Janjua, who left us recently, as well as Ali Asad, and the rest of everybody. Just want to say very quickly that we're very much open for volunteers and people to join our team. This is our email also, and also we're on Twitter, so please feel free to reach out to us. And thanks so much for your attention.
Thank you very much. Thanks very much, Dr. Moghreb. It's a pleasure. Thank you so much. Right. See if I can share this slide again. Are you able to see the slide with Dr. Saoud Al-Qazay? Not yet. Are you able to see the slide?
No, not the slide, not yet.
Not yet.
You can see your screen, Doctora. You need to put it into full screen, Doctora.
Okay. Right. Technology, hey. Okay. Right. So allow me to move on, because there's obviously some technical issues there.
You know when you share, there are actually two clicks. There's one click and then there's another one. So maybe just make sure that.
Not to worry. Let's move on if we can.
It's coming up now. It's coming up. We can see it.
It came up.
Okay. All right. Brilliant. Okay, so for our fifth speaker, we have Dr. Saoud Al-Qazay, and it's a real pleasure to have Dr. Saoud here today. He's one of our young stars, and I'm really looking forward to seeing him very many more conferences, hopefully in the future, and hopefully it will be more. It will not be virtual, and it'll be face to face. That we'll see you next time, because this technology I think is getting the better of me. So Dr. Al-Qazay has undertaken an undergraduate medical degree at the University of St Andrews, BSc Medicine, and at Edinburgh. He became interested in ophthalmology during his time at medical school and undertook a 4-week elective in ophthalmology. He then completed the foundation program in Oxford, in the Oxford Deanery, where he gained an interest in genotype-phenotype correlations in inherited retinal diseases when he worked at the Oxford University Hospitals. He is currently undertaking a Doctor of Philosophy focused on genotype-phenotype correlations in Stargardt disease and ABCA4 retinopathies at the University of Oxford. His career's aim is to become a clinician scientist in ophthalmology and subspecialize in vitreoretinal surgery and medical retina. I'm sure you'll assist him in joining us in the hospitals and working in the hospitals and researchers in the future. So please welcome Dr. Al-Qazay. The floor is yours. Thank you so much. I think you're muted. Excellent. Yes, we can hear you. Saud, we can't hear you, Dr. Saud. We're still having problems in hearing you.
So you're muted again.
Hello, good morning, everyone. Thank you for inviting me to do this talk today, Fatima. My presentation is going to be on genotype to phenotype correlation studies in inherited retinal conditions, giving an overview of what publications we've done in my group to date from the GISRA scholarship that I'm on. So inherited retinal diseases is an umbrella term used to describe a group of conditions that affect the retina with that are genetically predetermined. Today,
so do you have your slides? Because we can't see your slides.
Oh, can't see that. Can you see them now?
Yes, excellent.
So it's a group of conditions linked to over 270 genes that affect the retina that we know of to date. And we keep on discovering more of these genes as the research improves with genetic sequencing. These conditions are individually rare when we look at the specific genes, but as a group, inherited retinal diseases affect around 1 in 2,500 people. Diagnosing these conditions is complex because of the heterogeneous phenotype and genotype seen in these conditions. And to add to the complexity as well, some of these conditions can be syndromic, such as Usher's syndrome that can affect sight and hearing, and others are non-syndromic, where only the retina is affected. And there's a lot of variability in the ages of onset. So a lot of these conditions affect pediatric patients, but we can also see some of them having a later onset in adulthood. So it's very complex to diagnose these conditions, even with variability within this variance within the same gene even. The most common conditions that we see in inherited retinal disease are retinitis pigmentosa and macular dystrophies. Patients with retinitis pigmentosa tend to present with night blindness, so difficulty seeing at night and then darker conditions. And they also get a progressive constriction of their visual fields. So with time, their visual fields become so constricted that they can only see the central 5 degrees, which is like seeing through a keyhole. And even if that vision is considered to be 20/20, it's a very small field of vision, so it's quite disabling for these patients. And then the macular dystrophy patients, they tend to present with more central visual problems. So as we can see on the Mona Lisa, they have difficulty discerning faces, and they'll have a lot of problems reading, where they have missing letters and blurring of their central vision. And other problems that they'll have include problems of photosensitivity and taking longer to adapt from going in a bright environment to a darker environment, and that's because their cells aren't working as well. And some patients can also present with abnormalities in color vision, so that this will affect day-to-day life significantly for these patients. Phenotyping in inherited retinal diseases relies a lot on retinal imaging. So one of the modalities that we frequently use is color fundus photographs, where we take a photograph of the retina and look for specific features. And as we can see in this slide, the different retinal conditions can have different appearances depending on the gene. So that can help us ascertain which gene has caused the condition. And then we've got autofluorescence imaging, which is the black and white fundus image that we see here, and that can show us the general health of the retina. So in the second image in the middle, we can see the appearance of retinitis pigmentosa, where it's the rod photoreceptors that allow us to see night vision and give a decreased signal because the cells there are atrophic. And on the OCT image, which shows us the retinal layers, we can see that the center of the retina is preserved, but the outside layers are lost. So this gives us a good idea of what the retinal function looks like. And in the third image, this is macular dystrophy, more specifically Stargardt disease. And we can see that the region with the decreased signal is actually in the center, which signifies that the central vision is going to be affected in this patient. And similarly, on the OCT image, we see that there's loss of the retinal layers in the center of the retina, which would be consistent with central visual problems. In addition to the retinal imaging, we use the Snellen chart, so looking at what the visual acuities are, what the fields are looking like, and that just gives us an idea of the width, if they have tunnel vision, for example. Microperimetry more frequently used in research at the moment rather than clinical assessment, but that just gives us an idea of the retinal sensitivity. So when we're looking at outcomes for retinal for therapeutic trials and retinal conditions, this will become more relevant. And electrodiagnostic testing. So here we connect patients to all these wires. So it's quite a long testing procedure, but it gives us important information on what the photoreceptors, which are the light sensing cells, how those cells are doing and how they're functioning. So very important in the assessment of these conditions. As I mentioned earlier, the complexity in diagnosing these conditions stems from how variable the phenotypes can be and the overlap between the phenotypic appearances between conditions caused by different genes even. The inheritance pattern is also different in that we can see autosomal dominant, recessive, mitochondrial and X-linked diseases that can all have a lot of overlap in the phenotype. So it emphasizes the need to take a good family history and drawing the family pedigrees when assessing these patients. And then with the genetic testing, there's always difficulty in assigning pathogenicity of novel variants, for example, deciding whether a gene whether a gene is truly causative of the condition and knowing the natural history of the disease. And there's also limitations of the genetic testing of identifying whether the variant that we've picked up is benign polymorphism or indeed pathogenic. For pediatric cases, it's very important to identify these patients because they'll have severe visual problems that they develop with time. So if we identify them early on, then firstly, we can support them with their schooling, making sure that they have the items that they need, such as laptops to succeed at school, and benefit from having counseling as well. So at the Oxford Eye Hospital, we have a counsellor for children with this condition, and we've seen some of them with quite severe retinitis pigmentosa going to university, for example. Provision of visual aids, so whatever the visual problem that they have, we can support them with it, and that if they have problems at night, then advising them on using flashlights. If they have problems reading, then microscopes and telescopes and things like that, other equipment like that. Support groups, so with support groups, they can meet other people with similar conditions, and that can help them managing their condition and getting more information from the support groups on what's available as well. And in clinic, it'll be getting the diagnosis will be important because we're starting to develop therapies, and now for one gene in particular, RPE65, there is a gene therapy approved by the FDA and is also available in the NHS in the United Kingdom currently. Now I'm going to present some of the patients that I've written up as part of my PhD here in Oxford. And this is one of our pediatric cases that was found to have an NR2E3 retinopathy. So NR2E3 is a gene that's important for rod differentiation. So any abnormalities in this gene will mean that the rods do not develop in these patients. So they're born with night blindness. And what we can see in this 12-year-old patient is on the color imaging, there's a lot of atrophy in the mid-peripheral retina, but the center of the retina is well preserved. So, so we've got a good central vision, but this patient was having a lot of problems with at night from birth, and with time had very progressive decrease in her visual fields as well. I have a figure of the electrodiagnostic testing for the patients that we wrote up with this gene, and what we can find is they, compared to the normal controls, they have what we call an extinguished ERG, which means that the rod photoreceptors are not working, and so are the cone photoreceptors. But interestingly, their photoreceptors for blue light had a higher response than the normal cone response, which is consistent with this enhanced S-cone syndrome linked with this gene. So we described novel variants in NR2E3 in this paper based on that. And here we can see the visual field of the patient I presented earlier. So we have the central 10 degrees is preserved, but around it the vision is lost. So this patient will have a lot of difficulty managing day-to-day life because of the very constricted fields that she faces. Another gene that we looked at is circle as well, which is linked with retinitis pigmentosa. And this patient, although in the imaging is aged 50, him and his brother first developed problems at the age of 7, mainly with difficulty seeing at night and a gradual visual deterioration. And what we can see in the imaging is that he's now got widespread retinal atrophy with very little remaining retina left for vision. And this patient would only be able to perceive light. So being able to diagnose these patients early on at the age of 7, you can give them more information on their prognosis and have information on life planning, for example, and career planning. For when the vision deteriorates, if it's going to deteriorate to an extent of no perception of light, for example. And this slide just highlights the other patients that we wrote up with this that also carried variants in this gene. And what we can see from this is that there's a large amount of variability, and that the patient I presented earlier is very severely affected, but then we have more mildly affected patients in the cohort as well. So just highlighting the complexity in diagnosing these conditions. For my PhD, I'm mainly focusing on Stargardt disease, and this is the most common childhood onset macular dystrophy. And it's linked to variants in the ABCA4 gene and is inherited in an autosomal recessive manner. This is a very complex condition in that it has a very variable phenotype, but the characteristic features are that the macula is atrophic, that we can see flecks, and the peripupillary area, which is this area here, is spared of disease, and we can see that on multimodal imaging. And it can affect people, it can have an early onset before the age of 10, or we can also see patients with a late onset over the age of 45. But what's important about identifying the patients that develop it early on is that they usually tend to progress and have more severe disease compared to the patients that have a later onset of disease. And in addition to the complex phenotype, we also have complexities of the gene itself, and that it is a very large gene with a lot of variation, so it makes genetic testing also very difficult. And recently we've also found...
Dr. Saud, you just have just under 2 minutes left. Thank you.
And we found that even the non-coding regions in this gene can also cause disease as well. And these figures just show the variability in severity, where we can have very mild central disease or disease so severe that we don't have any retina left and we can see the sclera. And the autofluorescence also shows variable severity that we see. And this gene is important for coding for the ABCA4 protein, which prevents buildup of toxic material, which corresponds to the flecks that we see on the imaging. So we think that the more dysfunction of this ABCA4 protein there is, the more severe the disease is. So as sometimes we see retinitis pigmentosa with this gene as well. And it's highlighted by the significant phenotypic variability that we see highlighted by these 3 patients. And it has a high carrier frequency, so we can see it in families that are not even consanguineous, despite it being recessive. Although it's important to highlight that, for example, here we have a father that was affected at age 12 and has very severe disease. But then his son, who was incidentally picked up in clinic, had at the age of 37 had no symptoms, but we can clearly see that the phenotype did fit. And when we did genetic testing, we found 2 variants, and they segregated to each of the parents. So the importance of diagnosing these conditions is that, having a molecular diagnosis, is that it allows us to understand the natural history of these rare diseases. And this is important for counseling patients, and allows us to also provide information on the risk to offspring. But now with the research happening on gene therapy and other techniques such as optogenetic cell replacement therapy and other pharmacological approaches for some of the conditions, it's becoming important to identify these patients and have them ready for recruitment to trials once the trials become available. And this figure just shows a summary of the different therapeutic avenues being investigated for Stargardt disease specifically. These are some of the publications to date that have been linked to the GISRA scholarship that I'm on. To end this PowerPoint, I'd like to thank the Qatar National Research Fund for giving me the opportunity to undertake this research. And my supervisors as well, Professor Downs and Dr. Halford, who closely supervised me and given me a lot of guidance on genotype to phenotype correlation during my time in my PhD, and the rest of the team that I closely work with in the Oxford Hospital.
Thank you very much indeed, Dr. Saud. I have a bit of an echo. Excellent, thank you. And we look forward to you returning to Qatar soon, hopefully after your PhD. Don't stay any longer in the UK. Okay, so with that, I hope we still have Dr. Suhaila, Dr. Cugno, and Dr. Yunus on board. We have 1 or 2 questions. We're running short of time, so I'll have 1 or 2 questions for you if you don't mind. This is from our audience. Dr. Suhaila. Where can I go? Dr. Suhaila, we have a question. How much of the microbiome is static or dynamic by age, and how does that influence its reliance in developing diagnostics or prognostic markers profiles in pathological cases as IBD?
Yeah, thanks for the question. So the microbiome is known, the gut microbiome is known to change with age. So from 0, the date of birth, till 2 years old, when the baby is mainly focusing on breast milk or formula milk, the composition is different from when they start having solid feeding and then move to the regular diet that we all eat. Obviously, the type of diet that we eat also affect the composition of the microbiome. So we don't recruit kids. I mean, we don't have kids less than 2 years old, and we also take note of what is the type of diet that these patients have been on for the past a few months before being part of the study. So this is being captured. is already known that diet and age will play a role in the composition of the microbiome. The biggest bulk of the change would be between 0 and 2 years old. After 2, the composition of the gut microbiome will follow a pattern that is very well looking like an adult microbiome. Of course, different diet will cause changes. Use of antibiotics will cause changes, as well as other type of medicine or medication. As we grow older towards the elderly years, obviously there will be changes as well.
Thank you very much. We have another question. Well, we have several questions for you, but I'm afraid we run out of time, so I'm just going to have to move on to Dr. Cugno. Dr. Cuna, a question for you. Have you explored specific genetic immunomarkers in patients with spontaneous regression?
So, thanks for the question. Honestly, what we know, the spontaneous regression right now is only in Down syndrome, in the so-called transient acute myeloid leukemia. Can you hear me?
Yes, yes, we can.
So, and we know this is a trisomy 21 that is usually associated with a transient acute myeloid leukemia during the first month of life or immediately after the birth. And this is being extensively studied all over the world, trying to understand what's the cause for the regression. But honestly, we didn't have case like that in Qatar and in our cohort, and we didn't go deeper on this side.
Okay, we have another question for you, but again, out of time, so hopefully maybe we can post that to you. A question for Dr. Yunus. Have you investigated genes involved in DNA repair diseases to explain undiagnosed forms of neurodevelopmental disease, since some of the DNA repair diseases are associated with unexplained neurological abnormalities?
Yeah, thank you for the question. Yeah, definitely. So in our approach, in fact, we're not so much focused on specific diseases. So we use basically an unsupervised approach whereby we don't prioritize any panels. So because we have whole genome sequencing data, and the type of genes that we're actually looking up is mostly driven by whether or not they have variants that might look like pathogenic. Yes, so definitely the answer would be we're looking into those as part of the large context of all genes that we're able to profile in our study.
Thank you so much. We have a few more questions that have just come in, but I'm terribly sorry, but due to the lack of time, we can't address them at the moment. So we just need to move on at the moment. So we'll move on to the panelists. I'm hoping you can see my screen. Any chance?
Yes.
Excellent. Thank you so much. So it's been a real pleasure to have some of the most experienced physicians and scientists as speakers here today. But in addition to that, I have to say that we've thankfully have got 3 illustrious panelists who've graciously accepted to take part in our panel discussions and answer some of the pertinent questions to raise a profile of research in pediatric diseases and conditions in Qatar. And I'll say in no particular order, just the way that I happen to go through this. So it's a real pleasure to welcome Professor Ibrahim Janahi. Dr. Janahi graduated from King's Faisal University College of Medicine in Dammam, Saudi Arabia in 1992. He completed his pediatric residency at the University of Illinois, USA, after which he started his postdoctoral fellowship in pediatric pulmonary at Baylor College of Medicine in Texas and Houston, USA. After completion of his fellowship in year 2000, he returned back to his home country, Qatar, and started at HMC as a consultant pediatric pulmonologist. He established the section of pediatric pulmonary in 2001 and was acting as the director of Pediatric Emergency Center in parallel from 2001 till 2008. Professor Janahi is a holder of key and senior positions in the state of Qatar. In addition to his current responsibilities at CIDRA and research centers as a senior attending physician, division chief of the Pediatric Pulmonary Department, he also holds the position of chief of medical education and designated institutions of designated institutional officer. His leadership roles extend beyond Qatar to the region and internationally. For example, he is the founder and president of the Gulf Society of Pediatric Respirology and a founding member of the Arab Pediatric Pulmonary Association, APPA. Professor Jannah, his research is focused on precision medicine. He's authored over 70 scientific publications. Now, we also have a very warm welcome to Dr. Nazir, who is a graduate of Ruplandi Medical College. He completed his adult psychiatry residency at Michigan State University and child and adolescent psychiatry fellowship at the University of Michigan and Michigan, USA. He served as chief resident at both the above institutions. After completing his training, he returned to MCU and KCMS and joined the faculty of Department of Psychiatry in 2007. In 2017, he joined CIDRA Medicine at Division Chief, Child and Adolescent Mental Health Services. Dr. Nazir is board certified in psychiatry, in child and adult psychiatry. He has published numerous articles and book chapters. He is a co-author of Behavioral Pediatrics, which is now in its 5th edition, Clinical Aspects of Psychopharmacology in Childhood and Adolescence, which is now in its 2nd edition, and is an associate editor of Journal of Child Health and Human Development. He's written extensively on autism, spectrum disorders, internet addiction, substance abuse disorder, ADHD, schizophrenia, childhood anxiety disorders. Before joining CIDRA, Dr. Nazir was an associate professor at Western Michigan University and also at Michigan State University College in USA. Dr. Nazir's primary areas of interest include autism spectrum disorder, ADHD, and the interface between psychiatry and pediatrics. Next, it's a pleasure to welcome Dr. Khalid Fakhro. We all know him dearly. Dr. Fakhro completed his PhD in human genetics in Yale University, where he was part of a highly selective HHMI Translational Medicine Scholar Program. After 2 postdoctoral training fellowships abroad, he relocated to Qatar as part of the Department of Genetics Medicine at Warren Cornell College in Qatar. He subsequently joined CIDRA as a principal investigator in 2014 and became director of the Human Genetics Department, where he has built a robust genomic medicine research pipeline for the hospital. In 2018, he became the inaugural director of Precision Medicine CIDRA. He holds adjunct faculty appointments at Cornell and at Hamad bin Khalifa University, where he teaches classes and mentors the next generation of masters of PhD. In all honesty, the above short biographies doesn't really justify the experience and wealth of knowledge in our panelists and indeed of our speakers. But a very warm welcome to you all. So given the shortage of time, and I can see we have overrun. If I may, I will jump straight in and a few questions for you all. My first question is, obviously the experience of the coronavirus recession refocused policymakers' attention on childcare. In fact, the World Health Organization statistics by WHO focused on the human toll and the impact of the coronavirus pandemic, highlighting the urgency to accelerate progress to get back on track and recover equitably with the support of robust data and health information systems. This could go to Dr. Fakhro. What were the effects and impacts of the plummeting rates of admission for commonly studied conditions as a result of the coronavirus recession on pediatric research at CIDRA or indeed in Qatar?
Thank you, Fatima. Can you hear me okay?
Yes.
Okay. So first of all, I just wanted to thank QNRF as usual for taking the lead on organizing this seminar, and for your fantastic focus on pediatric rare disorders. I think Qatar can be a trailblazer on this on a regional level, and as we heard from the earlier speakers, the research that's being done both locally and internationally by some of our bright students as well is really quite promising. To put it very simply, I know we're short on time. I think COVID-19 had a very, very big impact on our ability to actually sustain research, specifically patient-focused research. And this was for two reasons mainly. I think the main reason is that all outpatient visits were pretty much suspended throughout all the medical institutions inside Qatar for a very lengthy amount of time. And so what that meant was that a large proportion of the patients that we commonly see were just not coming to the hospital. The other thing is that even for patients that were inpatients, for example, it was very difficult accessing them for research because of all of the extra restrictions, the PPE that's required in terms of patient contact. So, the hospitals took really extensive safety measures for the safety of both the students and the staff. Unfortunately, that had a bit of a toll on the ability to do research. But as we all know, QNRF graciously allowed a suspension of some of these projects, which was great. I really hope that now we can actually make up for some lost time.
Well, thank you. So what were the remedial actions that you took, for example, at CIDRA to help you in progressing your research? I mean, for example, were there alternative data collection methods, remotely, etc., that may have assisted you? What actions did you take?
Yeah, so many of the projects actually started focusing more on analysis of current data, generation of more omics from pre-existing collected samples. There were some innovations, for example, transitioning from blood draws to saliva or other types of samples. Focusing on some of our international collaborators where sample access was possible during this time. So a lot of these mitigations were project dependent, and I think as you know, many researchers are very innovative when it comes to finding ways to try to continue doing science.
So, Dr. Fakhro, what is the reliability or the validity of these forms of work that you conducted? I mean, these sort of remedial actions.
Yeah.
I mean, can you let the audience know that they are in fact reliable and valid?
I mean, scientists would ever do anything that would jeopardize the quality of the data they're collecting, or the level of science. So I think if someone felt that the quality was being jeopardized, they would just pause, right, and hold on to status quo rather than trying to come up with, as we say sometimes, garbage in, garbage out. So you never want garbage to come into your research. You always want to make sure that the collection is very high quality, the standard is met across the board. But unfortunately, during the COVID pandemic, patient safety was the #1 priority, and so we had to adhere to a lot of these rules.
Thank you. I've got one or two questions, but I'll leave it there. So if I may move on to the impact of the COVID-19 on mental health of children and adolescents, which was of great concern, particularly since COVID-19 is much more widespread than other epidemics such as the SARS. So I suppose my question is, I think hopefully to Dr. Nazir, it's been indicated that compared to adults, this pandemic may continue to have increased long-term adverse consequences on children's and adolescents' mental health. Therefore, much research is still required on the psychological impact of the pandemic on children and adolescents. So from your experience, what has been the impact of the epidemic on children's and adolescents' mental health in Qatar? You're muted. Can you unmute yourself?
Okay, can you hear me well?
Excellent, yes, I can.
Okay, great. Thank you, Fatima, for the introduction, very kind introduction, and the question. Yeah, so I think it has been challenging for every healthcare profession who are dealing with the child and adolescent population in the mental health area or in the medical area. I think this is one of the pandemic that really affected millions and millions of people all around the world, and it did not see whether someone is well placed in the society or someone is poor. It's just everyone. It's a blanket kind of like a problem. In our clinic, in the last 2 years, we had done a lot of different things to help the child and adolescent population, and we gradually see the cohort of the patients that we are seeing shifting from more the patients who are developmentally disabled, who are having school-related problems, and some behavioral issues at home, to a totally different cohort in which we were seeing more anxiety and depression. There were more and more late adolescent year patients that were coming in our clinic. And if you look what the pandemic has done, it has created a significant amount of uncertainty in our lives. The isolation, the quarantines, the loss of the jobs, loss of the financial productivity. We have seen kids in which the families are having a hard time even putting the food on the table. So just like their primary safety areas, they were just taken away. And of course, like governmental agencies and other people, they really help, but it is a very difficult a change, metaphorically and in a realistic way. That these children are affected. They started struggling with academics. There are a lot of kids who did not do well in the online schooling, not that they were distracted in doing maybe browsing or something else, but it was just not the mode that they gained anything. So the two things I think that we really appreciated during this COVID, the first is that we really do not appreciate enough what we have in our life. Until and unless it is taken away, we really do not know, and that's what we saw in the families and in the children. The second thing is that, and that was very happy things, is that before the COVID, I came here in 2017, and in 2 years, I think about 50% of my kids that we were seeing over here, they did not want to go to school because they hate school and everything is kind of like a very difficult and there's drama and everything. And after COVID, hopefully we are at the end of the COVID, and now we are seeing the kids in person, and I can tell you 90% of the kids want to go to school. And I never thought that I'm going to see this day in my life in which the kids say that, okay, well, I want to go to school. But again, this is just the perception. This is just the appreciation that what they have lost and how valuable it is to them, that is creating some resilience and positive outlook in the future. So that was some of the basic things that I think that I can say about the clinical side.
Excellent. So very quick question follow on from that. So what measures have you put into place, whether it be CIDRA or do you propose in Qatar, or do you recommend for access to mental health support in Qatar?
Yeah. So some of the things that we were done basically were in close collaboration with other providers, especially the Ministry of Public Health. I think they did a wonderful job in providing the education and also building a 24-hour national mental health line. So anyone can access that, anyone can talk about their problems and the proper referrals can be made. So whenever we talk about the pandemic or this broad scale trauma, the first thing always is the education, and Ministry of Public Health did a very good job. The second thing that we did is that we started getting involved very closely in the community. So for example, we wrote different opinion pieces in different newspapers or in the Qatar Foundation media. We also had a very robust ambassador program with the Qatar Foundation schools for the last 2 years. And the goal of that program was to take care of the stigma and to build some awareness around the mental health. And we start getting a lot of people who were reaching out to us from that. The kids were initiating kind of the referrals. Then the second thing that we did is that we reduced our waiting list really drastically. So for example, in the urgent cases, there was a time when we used to wait. Sometimes kids had to wait like about 4 or 5 days. Now we can entertain them within about 24 to 48 hours. So we also improved our internal processes so that we can provide the care to these kids in a timely manner. The third and the most important thing is that we stayed in touch with all of our patients, irrespective of they were acute or they were chronic. So when the COVID started, CIDRA IT did a marvelous job in building the tele health systems, both audio and video. And all of the divisions that I'm aware of, they jumped onto that and they started engaging. And the goal was really not to keep on providing the continuity of care, but just to tell them that, okay, well, we are here. And Dr. Fatma, you cannot imagine like how much appreciative responses that we get from the parents, that they knew that there is someone over there in CIDRA who can reach out to them at the time of the need and that they can talk to. So I think that was one of the most important things that we did. And our hope is that we will continue to provide the care virtually as well as in person.
Thank you very much, Dr. Nasir, for providing details of the effort that you put in, both yourself and the Ministry of Public Health. I've got one extra question for you, but I think I'll link that to the very final questions that I have for all of the panelists. My next question is, this could possibly go to Dr. Janahi, maybe you could address this. And we've been told that it's commonly thought, or indeed research has proven, that the origins of many adult diseases can be traced to negative experiences early in life. So confronting the causes of adversity before and shortly after birth may be a promising way to improve adult health and reduce premature death. What diseases have there been have their origins in early childhood?
Thank you, Dr. Fatma, for the introduction and the question, and thank you all for being here. This is very, very interesting and stimulating session. Now, to answer your question, there are a good number, probably almost a majority of the adult diseases that we see vividly in adults originates itself into childhood and sometimes even before birth. There are many diseases, like I can give you a few examples, like asthma. Asthma is a very, very common disease in adults and pediatrics, and it's probably the most common non-communicable disease in pediatric age group. That starts early on with a genetic predisposition to A2P and type 2 inflammation, and that could be triggered by exposures during the childhood, whether it's exposure to different viruses like rhinovirus, RSV, and some other viruses. We still don't know much about the long-term consequence of COVID in patients who are prone to asthma, what would happen to them in the later childhood and adulthood. But anyways, there are many other factors that influence the way that the disease would behave, whether it would show up its phenotype later on in childhood or in adulthood. In asthma, for example, if a child is exposed to these different triggers of asthma phenotype, including viruses, environmental allergens, aeroallergens specifically, and the role of the lifestyle is very important in determining and shaping up the asthma in adulthood. One very important factor that started to receive a lot of attention is obesity and the epidemic of obesity in childhood, and how that plays into the asthma phenotype as we move on. It sounds, and it's now well known, that this is a different phenotype, and that basically starts early on in infancy. Even so, obesity itself and obesity with asthma can be determined early on and even in infancy. There is a lot of interesting interaction between the gene and the environment that happens during the early years of life that would determine how we are going to end up living with or without a chronic disease. Another good example is pulmonary function, lung function. Your lung function is determined not only by your physical fitness or your genetics, but also how your lung growth happens because of these environmental and whatever you are exposed during childhood. Lung, for example, and pulmonary system in general keeps growing, especially lung, alveoli and all that, all the way until the age of about 8. And during that 8 years of life that you still are developing your pulmonary structure, these exposures and these behaviors, lifestyle, exposure to different environmental allergens, in addition to your genetic predisposition, might determine how lung function, how good or bad your lung function would look like in later childhood and adulthood. So the long answer to a very specific question is that, yep, the early years of life are very critical life ages that needs to be focused on in order to have health later on in your adult life.
Okay, thank you so much indeed. Now, my next question may seem a little simplistic, but just to it's a burning question. Do you know of any known treatments that can possibly help reverse the effects of early childhood diseases to try to circumvent adult disease?
Actually, I wish if there was a magic wand that you can use it in the childhood to prevent some of these chronic diseases. However, there are things that you can do. Lifestyle issues, lifestyle starts right there in the infancy, and that you, as a parent, you can start shaping up the way that your child is his eating behavior, his activity behavior, his interaction with the environment. Those are all amicable to modification during childhood. For example, and this is very interesting that some parents, they have this perception that they need to keep feeding the child, keep feeding him because they want to see a child who's chubby, and that is dangerous. So that piece is very much amicable to modification of behavior and the way the perception of health and ill health in children. And we obviously work with the parents to have this different, so modify, and this is something that, this is not a medicine, this is just kind of modifying the way that parents perceive their kids as healthy. Diet habits starts early on in infancy. Secondly, activity. Encouraging activity of the children to go out there, do their whatever physical activity that they might please, and parents supporting that sort of lifestyle area. Sleep, another one. Stress. All these are, I mean, I'm sure Ahsan will be happy with hearing this, but all this can be installed early on in the infancy and toddlerhood, and that can be carried on as healthy lifestyle throughout the rest of their life. There's one interesting theory which we always say, hypothesis, the hygiene hypothesis. So parents who are very keen on keeping everything so clean and so nice, and this maybe Suhaila would help me out with this too. So the more hygienious, quote unquote, you are in your infancy, the more allergic diseases you get. So that, again, another lifestyle issue. What is clean? What is not clean? All those interventions are soft, quote unquote, soft interventions, but are of detrimental effect in the long term.
So given that we've kept ourselves extremely healthy and clean for the past 2 years, we're now prone to major health issues in the years to come. Dr. Janahi, Dr. Ehsan, would you have anything to say on that?
Yeah, I think this is a really interesting topic, and we can talk a lot on this. Dr. Janahi made all the wonderful points. I think, Dr. Fatima, the main concept I think to think about is that the early identification and the proper treatment is the mainstay if we want to improve the life of the kids and the children and adolescents. 3/4 of the psychiatric diseases that we see in the adults, they have their roots in the childhood, and there are windows of opportunities in the childhood, starting as early as 18 months when we started diagnosing the autism, in which you can do a lot of different interventions. So by saying that, it is also important to say probably is that the mental health is not really, and the same as Dr. Janahi said about the asthma, these are not kind of like Manichaean type of like concepts. It's not really, there is no dualism, there is no all black and all white. These are all on the gray area, and depending upon the early you are able to pick it up, the more the better improvement you will be able to make. And that was honestly, that is something that people really need to understand about the child psychiatry, is that as simple a disease as ADHD, if you pick up at 6 or 7 years of age, you can just change the whole trajectory of the child. And if you don't pick it up, the same person will go into the substance abuse, into drugs, in the legal system, school dropouts, and it will just create chaos in his life and in the family's life. So again, early identification is very important.
Dr. Suhaila? You're muted.
Sorry, yes, I needed to add one more point to what Professor Janahi was describing regarding the developmental origin of health and disease, that while focusing on childhood diseases, it's also important not to forget that we need to take care of the pregnant women during pregnancy, follow her up systematically, because there are many studies right now that the in utero environment is really the first start of predisposition predisposing our kids to long-term chronic diseases and later adulthood diseases. So that's why we're focusing here at CIDRA to also implement research working on pregnant women, really assess how a woman with gestational diabetes follow up on the babies when they're born, on the first 2 years of life. So during pregnancy and the first 1,000 days of age are really important and critical to really make changes and try to avoid onset of diseases. One great example can be asthma, obesity, diabetes. All these diseases have a root to the in utero environment, and we know already that if we can change lifestyle or other markers during pregnancy, we can avoid or delay the onset of these diseases later in life for the kids. Going back to the hygiene hypothesis, yes, I agree again with Dr. Janahi that being too clean, and I'm probably one of these moms, but I keep the microbiome in my perspective always, because if we clean too much, we're killing those good microbes that are supposed to stay in a eubiosis or balance environment in order to protect us. So a lot of parents may be self-prescribing medication or antibiotics to their kids, or probiotics without any reason or condition. So these have to be well controlled. We need to keep in mind that there are these good microbes that are helping us with the digestion, they're helping us to keep ourselves in a healthy status, and any trigger or cause for imbalance is gonna predispose us to diseases. Example, asthma, type 2 diabetes, obesity, even autism has been linked to an imbalanced microbiome. So I just wanted to mention these 2 points, and yeah, thanks.
Thank you so much, Dr. Sahela. I'd just like to shift gear here for a while. My next question can be for Dr. Fakhro or Dr. Cuna. There's a well-known knowledge gap regarding the efficacy and safety of medicine in children of all ages, and children are often treated with medicines off-label, i.e. unapproved. And recent reports on the effects of legislation indicates that pediatric clinical trials remain a challenge. The strengthening of pediatric clinical research is essential to facilitate generating the data that will let children enjoy new medical advances in a similar manner as adults. So given the population size in Qatar, what challenges have you experienced in conducting pediatric clinical trials in Qatar, or indeed at CIDRA, prior or during the pandemic? Dr. Cuna or Dr. Fakhro? It's a bit of a loaded question. Sorry.
No, it's okay. I mean, I can just start and then I can pass it on to Kiara. But I think that you know, there are I would say two major difficulties. The first one relates to clinical trials. Is just as a nation we lack the right infrastructure, and I know that we have come a long way. And I think we, you know, regionally, I would say that CIDRA medicine, and I think the country of Qatar, is probably the most advanced in terms of biomedical research in the entire MENA, greater Arab world. We definitely have a vibrant ecosystem, brilliant scientists. We have a captive patient population. We have physicians that really care about transforming care for patients through clinical trials. But the infrastructure is still something that we're developing, and I think our CEO alluded to this earlier, and you know, Dr. Cuna can actually tell you a bit more about our plans to build a GMP facility to introduce cell and gene therapy, you know, on a national level. The other thing, of course, is the regulatory environment, and that's also something that's that has to catch up. You know, as I said, you know, we're a country that suddenly develops a lot of things all together, whereas the systems in place have still not been put into, you know, still do not facilitate the kind of vision that we're trying to get to. And there's actually been quite a few conversations on a national level with the National Academic Health System, you know, where CIDRA Medicine partners with the likes of HMC, Weill Cornell, etc., to really try to change policy on a national level, talking to the MOH, you know, talking to the Ministry of Education, talking to the Ministry of Finance, really trying to build this ecosystem that permits clinical trials at this level, because if we can actually pull it off, Qatar can become a trailblazer, as I said earlier, in terms of the ability to really attract the most complex patients and really transform the way that care is managed for them. And I think Kiara has a lot more to add on this. So I'll pass on the baton. Kiara?
Yeah, thanks, Khalid. I don't have that much to add. I want just to tell that we have to envision our ecosystem together with the other centers in the world. And we are experiencing right now, especially in my field, pediatric oncology and hematology, we are currently using on a daily basis off-label therapies that have not been tested on kids, and not because of the lack of efficacy, but because of the lack of clinical trial with enough number to justify their use. So we take on ourselves these responsibilities on a daily basis. And here, what's the role for Qatar? And this is also our thought in building our vision around clinical trial. So we have one of the most comprehensive biomedical research institute in the world, we can say, especially at CIDRA, but also in other, if we consider other institutions in Qatar. And we have to envision Qatar as a leading country, it's happening in several other fields for clinical trial in pediatric disease. Of course, we cannot start as leaders, but we have to start from running clinical trial together in the context of network, a well-established network with Europe, with USA. And this is exactly what we are trying to do right now, to build the infrastructure that, of course, will be an infrastructure that will be able to support clinical trial from those that are simpler, where we are joining other leading institutions, towards our leading function maybe in the future. But for sure, the infrastructure is the first, and the regulatory environment are the two mainstays for helping our kids here in Qatar, but also exporting all our knowledge abroad.
Okay, Mabi, thank you so much indeed for that. Due to the lack of time, I just need to move on. I'm trying to get as many questions as possible in while I have you here. My next question is, due to the lack of top-level clinical research positions, not least due to the great technological advancements over the years, the British government, for example, has recently urged to help finance a creation of new nationwide schemes of tenure-track clinical scientists intended to make research careers more attractive to qualified physicians. What measures do you propose should be taken to address the lack of critical mass of clinical scientists in Qatar? And that can go to anyone, whether it's Dr. Janahi, Dr. Fakhro, or Dr. Nazir.
Let me take a stab at this, Khalid and others could pitch in. So this has been one of the...
Am I correct to assume that, Dr. Janahi, that we do have a lack of critical mass in sort of physician scientists?
Absolutely. That I would, if you ask me in Qatar, what is your single most challenging stumbling block to progressing clinical research, I would say not enough mentors. And therefore, and those mentors need to be protected. They need to be really supported. And one of the critical support of these mentors is a career path for their trainees. So unfortunately, although the career path for clinicians is very clear and kind of predictable, you do your internship, residency, fellowships, and you're on your way to become an independent senior attending, an attending and senior attending. While that is not well established in Qatar, maybe in some other more advanced, I would say more mature centers, countries, they do have a pathway for the clinician scientist. We don't have this here. And I think that this is something that needs to be tackled at the top level. So it is not bottom down, it is top down. And therefore, there has to be I would say, a will and a mandate from the top leadership that, we are now recognizing a path for the clinician scientists. They need to start, we need to start way early on during their medical school. I feel that we still need to have a well-established MD PhD program, which we don't have that here. We only have either MD or PhD. So that is either you are a basic scientist, you will be struggling as you move on to find grants, and it's going to be very challenging for institutions like Sidran, Hamad, who is primary goal is basically clinical. And therefore, these basic scientists and translational scientists will have to really tag along with clinicians, and not all clinicians have the orientation and have the understanding of importance of doing their practice on a scientific basis. So I hope that I'm trying to make myself clear, but I hope that I'm not confusing people here. No. People, residents and fellows who are on pure clinical path, they do not appreciate the rigor that is required to have a scientific base to their practice. And that on the other hand, scientists who are on pure science, they look for cytokines, genes, exosomes, and so on and so forth. They do not appreciate the loads of challenge that clinicians have. The solution, in my opinion, it starts early on by setting up a pathway, starting from medical school, an MD PhD program that could be integrated into both clinical and the basic science together. So therefore, the clinician scientist mentality, the clinician scientist practice will be cultivated and supported. This is, in theory, this is great. And people in the last 10 years that have been having this discussion, at least 10 years that have been having this discussion at different levels in the country, everyone talks the talk. No one is ready to walk the walk, because walking through this, it requires a massive amount of paradigm change. Start with regulation, funding schemes, recognition of effort and recognition of this path as very important, as critical. Now, today we have seen how important it is to have clinician scientists, how important it is to have scientific base to all the practices. When Khalid Hussein mentioned how diabetes management in children has really been transformed by being a scientist clinician, not just being a practitioner clinician. And on the other hand, and we've seen examples of all these different diseases, and this is not unique. There are many, pretty much all the diseases. If you start kind of dissecting it, you will find that there is a bad need for people who are clinically savvy, but also very much scientifically savvy as well. So my advice would be that we need to, since Qatar is now trying to build itself as a center for the next generation of precision medicine, other things, we need to have some basic building blocks taken care of. Regulation, recognition, creation of these pathways, those are critical. And I can't wait to see that happening. It has been one of my personal dreams and my colleagues' dream to have this pathway nicely mended and nicely clarified, things that would attract people to go into the rigor of research instead of doing a classic clinical pathway. Is the reward that is associated with it? Is the recognition of that? And so on.
No, absolutely. I wholeheartedly agree. I can see that this is a passion of yours. Dr. Saud, would you like to chip in on that? Because I know we had a little discussion about this earlier. Please go ahead.
Yes, so the clinician scientist pathway is actually the one that I would like to go through. And in the UK, as you mentioned, Dr. Shinahi, that in medical school they have although they don't really have the MD PhD program in the UK, it's rare for people to do that. They have an academic training program after you do your medical school where you get some protected research time, and there's junior fellow jobs usually along the career pathway that have protected research time that have worked quite well within the NHS, to my understanding, and I have friends who have done their PhD and have gone on to do some of these academic pathways, and they find that having that protected research time actually helps enrich their research experience, and they benefit more from knowing that they have that time compared to finding the time on their own. to do their own research alongside having a very busy clinical job.
Okay, thank you. But you can see, obviously, that from our support of Saud, that we totally wholeheartedly agree with you, Dr. Ibrahim. So now I'll again, due to lack of time, I just need to push on. My last question, and it again is quite a loaded question here, and no matter how much data or research or papers you may read, the question here is, what are the research gaps in childhood diseases and conditions specific to Qatar? Now, as I say, if you read through various publications, etc., it may be specific to a particular country. Do you feel that research gaps currently in Qatar are the same as or different to others, or to the needs of others in other countries? I can go to first Dr. Fakhro, Dr. Nazir, please, after. Dr. Fakhro, for you first, please.
Thanks. So we are in many ways a startup, as our CEO mentioned earlier. So we are sort of at the beginning here. And so if you want to talk about gaps, I would talk about building out a strategy that really over the long term positions us well to be able to address and implement research for every patient that walks into our hospital. So it's not really a gap per se, it's just something that we haven't developed yet. It's not something that doesn't exist because of any other reason than we are a startup. But I would say probably one of the biggest gaps in the Qatari health system that impacts the ability to do good research is a very poor integration between medical records and research records. And that's something that we've identified very early on. Where the systems are still very much siloed. Even the systems at different hospitals here or different private clinics are very much siloed. And so the ability to really follow up a patient over the course of their lifetime becomes incredibly challenging. You asked a question earlier about whether impacts in childhood are going to affect adulthood. I think one of the most exciting things about Sidra Medicine as an institution now that we're open is the fact that, again, as our CEO mentioned earlier, every child in Qatar is going to come through here for one reason or another, right? And so we'll be able to start building those databases longitudinally. So we have to really start thinking about, the 30 to 50 year time frame. That's the kind of what do we have to do today in terms of building repositories, in terms of proper tracking, in terms of what biomarkers do we need to produce, right? There's always a cost benefit estimation of how much is it going to cost us to do this at scale versus what's the benefit going to be in the future. And so we constantly discuss this amongst ourselves, with our clinical colleagues. We also discuss it with our international partners in terms of best practice. But our main question is, can we start very early on in terms of detecting the potential for disease and really building, identifying our gaps early, in the sense that in 30 years we're going to have a major problem with obesity, diabetes, stroke, etc. Or should we be focusing more on neurodevelopmental conditions, autism, or some congenital conditions, etc. So we won't be able to map that out until we actually start collecting that information, integrating it properly. So the only place that we have started doing that systematically, and I think this is it would have been in no way possible without the will and the vision of the Qatar Genome Program, has been to start at genomics, to start by building a genome facility here, sequencing every sick kid today, but eventually probably every kid that comes here. The reason we do genome first is because your genome is constitutional. It's there with you from birth. That's not going to change, right? So at least you can start doing that at the baseline for everyone. And I think our partnership with Qatar Genome, with the new Qatar Precision Medicine Institute, really thinking about this on a national level in terms of what to do. The beauty of doing research in Qatar is that you continuously discover gaps and you continuously learn and you continuously improve. I think that's why it's such an exciting time to be here and it's such an exciting time for research.
Thank you, Dr. Nazir.
Yeah, I was thinking about that, these gaps. I think they're specifically talking from the mental health point of view. I think there are gaps kind of all around the world. I don't remember there were any really federally funded studies started coming up in the US before 2002, 2004, when we went into the these antidepressant medications and the black box warnings. And before that, what Dr. Fakhru said about 20 minutes ago, it was exactly that. It was garbage in and garbage out. And that was really, you were not really getting any kind of a consistent data to base your clinical decisions on. So in Qatar, I really think that we are at an interesting place. And there are a few things I was thinking about what Dr. Fakhr was talking about. First, I think we really need to have the prevalence studies. There are some that recently came up. There's a prevalence for ADHD came up, for mood and anxiety came up, for the autism came up, but we need to have like a much more detailed and much more population-based prevalence studies. The second thing I think that is really interesting in this country is that the type of patients that we see, they come from about like 150 to 160 different nationalities. So looking at the cross-cultural phenomenology, at least in the mental health, that is in the clinical side, that is just amazing. Like a different clinical disorders present in a very different way, depending upon their cultures, depending upon their beliefs, depending upon what the parents think and how do they handle themselves, how do they handle the child. And the third thing I think what Dr. Fakhr and Dr. Junahi has already elaborated on is the clinical trials. And also the one thing is building the normative data. So for example, in our field, having the rating scale is really important. Let's say if I want to diagnose someone with ADHD, I will do it on the Conners rating scale. But when I compare the results of a 6-year-old Qatari child, the only normative data that I have is from the US-based population. And that doesn't make a lot of sense that, okay, well, yeah, I can do that. But again, there is so much cross-cultural variation that we can say, yeah, you are ADHD, you are not. But in the back of our mind, we all wish that, okay, well, we need to have more normative samples in this population. And I think those things, at least from the clinical point of view, that will take our field significantly forward.
Lastly, I'll give the floor to yourself, Dr. Junahi. Would you like to add anything to that with regard to knowledge gaps or research gaps currently in pediatric diseases and conditions in Qatar?
Yeah, I mean, I definitely agree with Khalid and Ahsan on what they have already elaborated on, and I can only add that there is place to fill in so much gap that we already have. I could speak about the respiratory diseases because that's my specialty, and there is still a huge knowledge gap in regards to common diseases, but also the more rare diseases that are more common in Qatar and in the region because of consanguinity, i.e. the monogenic diseases. So there's a lot that we need to learn about when it comes to pediatric diseases in general, but also respiratory in specific. And I would say asthma is a good example of knowledge that Qatar can be unique in trying to provide the world with the genotype-phenotype relationship, given the fact that it is a very contained cohort of patients, but also the ability, the infrastructure that we already have to deeply phenotype them and also to genotype them. So therefore, opportunity is there, and I think we can keep just jumping on these opportunities and providing more and more of our interest, our time, our effort, and build these capacities like we are seeing there to be able to really fill in some of these gaps.
And I'll just give the floor to anybody else, Dr. Suhaila, Kugno, or Dr. Saoud, before we close off. Anything from you? Dr. Suhaila?
Yeah, I think I echo my colleagues here that, I mean, I may be summarizing what everybody else said, that to be able to address these gaps in pediatric research in Qatar, it is really important to understand the landscape of these diseases here in the country. And to be able to do that, we need to identify the genetic and non-genetic determinants of a specific disease in the pediatric population. And because it is not feasible due to physiological differences, we cannot kind of understand what is happening on the pediatric population from our knowledge on the adult population because they are already physiologically different. And we cannot assume that the same thing is happening from our knowledge on the pediatric diseases in the US or UK or Caucasian population, African population, because there is no much knowledge or research done on a pediatric population here in Qatar. So I echo how important is the work that is being done by Qatar Genome Project. I am hopeful, inshallah, that with the outcomes, we should be able to develop a better, for example, screening system for the newborns of the Qatari population. We should be able to understand more of the genetic and non-genetic causes of the disease. We know for sure now the data is directing us 33% of our kids are obese. So this is a big problem. We need to enrich our resources in obesity research, try to modify the lifestyle, try to work with the clinical team on how we can understand the reasons and really work with our kids, probably the moms before getting pregnant. I go back to the Doha paradigm because obesity is one of these conditions linked to the in utero environment. So these are different gaps from different perspectives, but also if we work together at a national level, inshallah, we should be able on the right track to address them and start implement more research on the pediatric diseases. Thanks.
Dr. Cuna, lastly to you. This is last guest.
Yes, I would like to add one concept, because I'm afraid that the national level will not be enough for pediatric disease. And so we are not facing, especially for clinical genetics disorder or cancer or I don't know, autism or ophthalmological disease. We don't have enough number to come up with reliable data. We are experiencing for lots of our kids extremely high toxicity to the drugs that are commonly used in US or in Europe. So there is something different. And if I believe that we should, at least this is my personal opinion, create a local network, joining us with the neighboring country, looking at Arabic ethnicity, and maybe, so this is something that we still miss. Maybe it's present in some fields and some other, and not in some other, but only working together in the region, we probably will be able to come up with reliable data that we can generalize to our population.
Well, thank you. You're quite right in that, because more often than not, sometimes when we get our proposals and we have our reviewers who look through them, we constantly have these comments such as, well, what is the incidence of so-and-so disease in Qatar and why is there such a focus on this? So I totally understand that essentially the numbers are not high enough for us to conduct much research in pediatric anyway. So we need to sort of broaden our horizons and cast our net wider afield. So with that, I thank you so much indeed for being gracious enough to stay until the end of, I think we've run over by half an hour, but at least we started a little late. I thank you so much and hope to see you again with sort of in-person conferences in the future. Thank you indeed to you all. Thank you so much for your time. All the best. Okay,
thank you.
All the best. We hope to see you back in Qatar soon, Dr. Saud.
Thanks. Thank you.
Thank you.
Thanks for inviting me to this.
It's a pleasure. Bye for now. Bye-bye. All the best.